So hello again. I want to welcome everybody back after our poster session this morning. Thanks for all of you who participated in that, and thanks for all of you being here this morning. Just very briefly, people always want to know some stats. So here's our stats. Total number of registrants that we had this year was 496 people, which is just under 500, and as you know, we usually max out when we fill our auditoriums 350 people. So we did get more registrants. I'm sure some of them did not actually come, but that's the official number. We had 77 presentations this year, which is pretty typical for most years. We had one keynote speaker, 24 10-minute speakers. We had 27 3-minute talks. We had 25 posters, and the stats that I always like to look at too were of those 77 presentations, how many were made by people who never participated before in the symposium. And the answer is 33. So just under half, about 43% were new folks. So that's one of the things we really like, is to always have new people, mostly students, but not always. We get a lot of new professionals who start working on the lagoon as well. We had somebody who joined us from Washington, for example, Washington State. Also, we like to look at how many of those presentations were students. And the answer is just over half, which is pretty typical. Presentations that were by students this year. I want to thank everybody again. This will save time later in the day. I want to thank the great steering committee that we had. You've seen them in the various sessions as moderators. Some of them are working behind the scenes, heading up the judging effort for the students. It's a really important part of our effort every year, as I mentioned yesterday. I want to thank our sponsors again, our founding sponsor and lead sponsor, Harbor Ranch Oceanographic Institute Foundation, and also the Indian River Lagoon National Estuarine Program, and all the folks that we recognized on the program, and I mentioned that a little bit more yesterday. I need to share with you that we have had a great team working behind the scenes. Sometimes when we meet in person, you get to see them, but usually even then, they're behind the scenes. Most of the work is actually done as part of our Indian River Lagoon Observatory effort, and in particularly this year, and so they all have regular jobs besides this. So this year, especially Kristen Davis, she's our Erlon manager, and you'll actually hear a presentation in a few minutes by Kristen. David Uberger, who is a biological scientist here in my program. Haley McQueen, who her day job is she's our lead water quality analyst. And Brian Botson, and his day job is he is our lead data analysis and also website expertise, which you again will hear about in a few minutes. Also this year, because of the virtual aspect, we were really happy to have strong support by our IT group, in particular that effort that was led by Wyatt Hudo and Nelson Beeman, who is the head of the IT group, and lastly, they all really helped out quite a lot during the course of the event. Last year, we started something we call remembrances. And it was the first year we had done that. And we said, we would do this again if we had a similar kind of situation. And basically what we're trying to do is to recognize that the passing, unfortunately, of some of the folks that have contributed a lot to Indian River Lagoon research and management. And this year, we do want to recognize Leroy Cresswell. So Vincent, I think you can share your screen now, and I think it would be okay. Can I just tell you, I wasn't going to really say anything about Leroy. I was going to let Vincent do that and maybe add something afterwards, but Leroy was really special to many of us. And I don't know if Vincent will mention this or not. It got mentioned in the program, Leroy first came to Harbor Branch in 1983, and he came to work with John Ryther. And John Ryther had built really a nice aquaculture program here. And that's when I first met Leroy, because I was also part of that effort. And
Dennis, can you hear me?
yes,
Okay, good. Sorry about that. I'm trying to share my screen, but it's not. There we go.
yeah, there you
It
go.
was stuck in a loop for a really long time.
Okay.
There we go here. Sorry about that, everybody.
All right, go for it. I was starting to tell some Leroy stories to give you some time.
Yeah, thank you. Can
I'll say, let me just finish up so I don't add anything later. Um, Leroy did a lot of things, and when he left Harbor Branch, it was wonderful that he had like a whole nother second career for many years with Sea Grant, and you'll hear more about that. But I will say that when he came on and joined our steering committee, the thing that he embraced the most was giving the students the opportunities and the awards. And the last time, and so he was always on that subcommittee. The last time I saw him was about a week before last year's symposium. He came to my office because every year he would provide a lot of the certificates himself. And he always wanted to make sure everything was just so right. He came in with a whole box of stuff. He gave me the box. He said, my doctor says that I shouldn't be coming to meetings because, my immunity is being compromised. And I knew his situation, although this was before his final crisis. And when he left, I knew that was probably the last time I was going to see him. But he wanted to make sure that everything was right, and he did. Okay, you ready?
Yeah. Sorry about that, everybody.
That's
Dennis,
okay.
can you see me? Because it looks
Don't
like
worry,
my
we
cam.
see Leroy. Don't
Okay,
worry.
that's all right. Cool. Okay. Yeah, thanks everybody. And yeah, as Dennis said, we'd like to remember Leroy. And you can follow his career arc in the remembrance that's in the agenda. But I just wanted to mention that, behind that list of lengthy accomplishments are these wonderful stories. And oftentimes they're very humorous that he's shared with me. Several of you here heard more of his stories than me. Some of you were in those stories. Maybe you, Dennis. You know who you are. From that wonderful history, I take away two things. And he told me just some really great stuff and really funny stuff. I take away two things. One, it sounded like to me, being a marine scientist in the 70s and 80s sounded like an awful lot of fun. Maybe a tad dangerous with maybe a touch of hedonism. For those that were there, like I said, you know who you are. And two, Leroy had a deep respect and appreciation for his colleagues and mentors. He recognized that he stood on the shoulders of some greats, people like John Ryther, for example, his early boss at Harbor Branch. But the one thing that I really got from him is that, with he had this great respect for those that came before him, but that was equaled by his encouraging those after him, especially students. I think he was as appreciative of the future as he was of the past. And if you stand on the shoulders of those before you and hope that the next generation will stand on yours, you have to realize that they need a boost, a hand up. And Leroy was that kind of guy. Whether it was personally welcoming new agents to Sea Grant, ringing the bell at each Gulf and Caribbean Fisheries Institute meeting, and engaging the many students at conferences like this one, or welcoming me when I first moved out this way. That's what I remember the most, his welcoming spirit and dedication to fostering that next generation. To that end, Leroy was deeply involved in student judging and support here at the IRL Symposium. And it's only fitting that we name one of the student awards after him, the Leroy Cresswell Award for the Best Student Poster. And for me, right now, that's a perfect segue to implore all of our judges to submit their scoring sheets on time. We have a very small window to tabulate those scores, so please get these to us via the Google Drive link that's provided. if we were meeting in person, I can picture Leroy running around collecting scoring sheets. I'm kind of doing that now virtually, but I'd like to think Leroy is with me as well, although spiritually. Anyway, thanks a lot, Dennis. Thank you for giving me the opportunity to talk about Leroy and give us a chance to recognize his contributions to the Indian River Lagoon. Thanks.
Thanks so much, Vincent. I think Leroy would have loved that. And now actually, Vincent is actually our next moderator, our next chair. So it will be back to him and he'll take us right into move things right along.
Yeah, are we ready? Are we ready to go?
We're ready.
All right, great. We're a little bit behind schedule, but that's okay. So now we're going to start oral session 3, water quality and human impacts. And our first speaker is, as Dennis mentioned, is going to be Kristen Davis. She's going to talk about the Indian River Lagoon Observatory Network. Go ahead, Kristen, when you're ready. Let me stop sharing. There we go.
Oh, thank you for that introduction. So today what I'm going to be talking about is our Erlon data portal, which was part of our Erlon 2.0. And if you attended the poster session earlier, my co-author Dennis summarized the other tasks for Erlon 2.0, but I'll be covering the data portal function. So the data portal was launched in August as part of Erlon 2.0. It's comprised of 2 parts, Linode Cloud Server, which hosts both the Linux LoggerNet, as well as a custom Postgres relational database, which facilitates and houses streaming data. The second component is a public-facing website that provides users with the ability to view interactive maps and plot and download real-time data. The data portal has a series of tabs across the top of the page, with each tab providing additional information about Erlon. The homepage provides a brief description of Erlo and how to access and or view the data, as well as a map showing all of our current sites. The About tab at the top provides additional information about the network, as well as the Indian River Lagoon Observatory. Finally, our newest tab is the QA/QC tab, and this provides additional information about the quality assurance and quality control procedures that are employed by Erlon. This includes a description of the real-time QA/QC procedures that we want run on all of our incoming data, and which we use to generate the summary flag that is used for visualizations and is included in all of our data downloads. As part of the map functions of the data portal, users can select to add a radar or tropical cyclone layer to the main map. This feature is available on both the homepage and the individual station pages that I'll show. And the radar imagery is from NOAA and is an animated feature. Similar to the radar function, we also have a tropical cyclone feature that you can add. And this will show the predicted path of any of the named storms within the viewing extent of our map. And the example I'm showing here is just what the predicted path was for Hurricane Isaias on July 31st in its proximity to our stations. The map is available on both the homepage as well as the individual station pages. Each site is represented by a circular icon here, and it's based off of, it's cut into different slices based on the number of related stations. So at each of the Erlon stations, we have 2 different types of stations. We have a weather station that includes all of our meteorological sensors that are above water, and our water quality stations that are all of our below water instrumentation. Now, all of these are located on the same ATON piling, but we've separated them for ease of view on the website. When on the homepage, if you hover over one of the slices of our sites, you'll have a snapshot of the most frequent or the most recent data from that station. The user can then go to a station-specific page by selecting from the list on the left-hand side of the screen, or by selecting from one of the circular icons. Each station page is going to include the station map that you can zoom in or out of, a box that includes all of the most recent data for that hour, as well as a figure for each parameter that's listed in the observation box. You can select your time frame in which you want to look at the data. You can turn a quality filter flag on or off. Parameters can be shown either in metric or English. And when you click on the station information button, it will give you a pop-up window that will describe the station as well as the date that the station was deployed. So the quality flags that we generate from our real-time QAQC procedures will be used to filter, can be used to filter the data using this function here. When the quality filter is set to off, users have access to all raw hourly data, and any data that's flagged as suspect will be shown with a bright X here. So this top figure is just showing the raw data for dissolved oxygen for a week at a time. But you can see we have this obvious outlier here that is causing the data, the rest of the data that is flagged as good, to be misrepresented as a flag, as a flat line. So once you click the filter to be on, you'll see the rest of the good data is plotted. So another part of the data portal enhancement that we wanted to make was to facilitate how users can better understand the data that we are generating. So in that sense, we created two different types of flags for health indicators. The first is a reference flag. And so this is the same figure from before that has the quality flag set to on, but our health indicators are set to off. Turning the health indicators on is going to give you, the health indicators reference flag on will give you levels for key water quality parameters, including stressful conditions. And so for dissolved oxygen, we're showing as anoxia, hypoxia. And the little icon here, it'll give you a definition of what we consider that condition. The other type of health indicator that we're looking at is a summary filter. And so this is going to give you, once you click the summary filter, this will give you lines representing the wet season, dry season, and annual mean of all the data for that parameter at that specific site. So for dissolved oxygen, you've got our wet season mean, our annual mean, and then our dry season mean. And so for this specific time period, you can see that we actually have dissolved oxygen levels that began above the dry season mean, quickly came to the annual mean, and we're approaching our wet season mean, even though we're here in the dry season. Similarly to the reference filters, if you click on an icon, it'll give you a definition of what that parameter means. And when you want to download the data, you can go back to your station page, click data download, and you'll have a pop-up box here that will allow you to select which single parameter you want to download, the data range, the time zone, units, as well as the format. And you can see we have several different formats for data download. Once you do that, you'll need to log in to access the data. And if it's the first time that you visited the website, you'll have to do a one-time free registration for the website. Once that's complete, data will appear in the format that was selected and include the following information: our data disclaimer statement, as well as how to cite any data use for for the data we generate, a description of the site that's the same as what's on our station page, as well as the data itself. And so this will be your timestamp, parameter, and that related quality flag. Now to view multiple data parameters and our data from multiple sites, you can use a query builder. And for that, you'll go to our download data button, function. The pop-up window will come up, and then you hit query builder. So this will take you to a new window and allow you to select multiple parameters, as well as a date range, and allow you to download the data either as an image or as several different types of data downloads, either a text file, a CSV file, or programmatically in a GeoJSON format for machine-to-machine data ingestion. All of the data downloads include the same information that is described on the previous pages. But overall, this is a living website. We really are looking forward to adding additional information to help for data access, data understanding, and we will be continuing to do this frequently. So please stay tuned. As Dennis mentioned in his presentation, we have an amazing technical support team. So I'd like to acknowledge both them and my co-authors for the support for this effort, as well as the website support by Carlton Gavarina, and then as well as our funding sources, which is the Harbor Branch Oceanographic Institute Foundation and the Save Our Seas license plate that's administered through the Harbor Branch Oceanographic Institute Foundation and the Florida Department of Environmental Protection. And I'll be happy to take any questions. So are there plans to add more sensors, specifically in the northern reaches of the IRL mosquito lagoon? Actually, this is some part of Erlon Point 2 that was covered by Dennis in his presentation. So you can get our exact sites if you get to see his poster. But yes, we are planning to expand into Brevard County within the next month, hopefully. And we're adding 2 different sensors, a PCO2 sensor that will be combined with our CFET sensor that measures pH, and that will allow us to estimate the rest of the carbonate parameters, including aragonite saturation. And then we're also adding another fluorometer designed by Kearney that will allow us to measure phycocyanin and phycoerythrin.
This is Dennis. I think I'll just jump in, and if Vincent comes back, he can continue to chair. I'll take the draft up here. Dakota, you should be able to share your screen now. Our next presentation is by Dakota Lewis from UCF, applying an assembly modeling approach to understand shifts in the estuarine fish community following a large fish kill in the Indian River Lagoon. Thank you.
All right, so oftentimes algal blooms, non-toxic or toxic, as they break down, can lead to differing hypoxic events and other anoxia. These hypoxic events can also lead to large-scale mortality events and fish kills. Following these fish kills, we can also see shifts in community dynamics and shifts in trophic linkages. So my main question for this talk is what happens to the relationships among sport fish, forage fish, and their environment after a complex disturbance. So there was a large fish kill in Banana River in March of 2016. If you look to the figure to your right, that's showing a hotspot map for the fish kill hotline reports. And so that's kernel density, just looking at likelihood rasterized. But I also collated other large data sets, mainly using the fisheries independent monitoring sampling, large and small seines. Those green dots indicate biotic samples that were used over the course of the study with both baseline years and the disturbance event. I also was able to utilize the St. Johns River Water Management District continuous water quality data, and there was a great monitor that was located right at kind of the epicenter of where we saw those reports coming from. So this is that continuous water quality data with the metrics on the y-axis and then the dates starting in fall of 2015 to winter of 2017 on the x-axis. You can see the bloom right there, which began in about mid-December in 2015 and lasted through mid-March of 2016. This steep chlorophyll die-off led to hypoxia to occur in the Banana River, subsequently leading to the fish kill. And so this is a breakdown of the fish kill hotline reports, where on March 21st, when the hypoxic event occurred, we saw a spike in the frequency of reports. For this talk, though, I'm going to be focusing on those 3 months post-kill, so April through June, and then comparing those to the same month, April through June, in baseline years. So first to start, I looked at, I'm interested in the linkage between the sport fish and the forage fish or prey that we were looking at within the area. So the sport fish here were captured in 5%, so it's really focusing on those higher abundance in this specific region: red drum, sheep's head, snook, black drum, spotted sea trout, and ladyfish. And then about 50 or so forage fish that were lower trophic level and present in the region. So as you can see, with abundances on your y-axis and months on your x-axis, the sport fish didn't necessarily get a large hit in their abundance that year during the kill, but they did have recruitment issues in subsequent years. Forage fish took a pretty big hit and stayed relatively low compared to baseline years in their abundance. However, the question here isn't necessarily what caused this drop in abundance. We know there was a fish kill, we know there was a hypoxic event, we know there was a large algal bloom. It's what happened to the relationship between these species and their environment right after that large event. So to answer this question, I will be utilizing Bayesian structural equation models. So structural equation models are made up of latent variables which represent a key component of the system, such as water quality. And then this latent variable is comprised of multiple observed variables or measured variables like temperature, DO, pH. So then using all of those different metrics, you create a saturated model, and these are comprised of all quantifiable metrics within your data frame. However, to make more parsimonious, these can be compared to more parsimonious or nested models, which have less variables in them and sometimes make it easier to understand more fine-scale relationships. Those are compared through information criterion. The lower the number, the better comparable fit. You can also compare the fit of the model. You can look at the individual fit of the models through different fit indices. I'll be using Bremsey, and in ecology, the numbers are kind of up in the air as far as 0.1 or 0.08, but the lower the number, the better the individual fit. And today I'll be using 0.1 as a good indicator of individual model fit. So when we're looking at the potential shift in these relationships, the figure on the top is that baseline saturated model. So these include all data from April through June of the baseline years in the study. So 2012, 2013, 2014, and then 2015, 2017, and 2018. This model has decent fit based on the Bremsey indice. And then that bottom model includes another latent variable for just that time post-kill, and included bloom dynamics. This model doesn't quite show great fit. And so sometimes it's necessary to look at more nested models to really understand what's going on within the system. So these are the nested models which utilize a framework to get to the construction of these. But the top model is that baseline, more parsimonious model. It still has decent model fit, and it also has lower WAIC and LU scores, meaning it's a better fit than the saturated model. That figure below also is better fitting than its saturated model with that just post-kill data. But what I really want to focus on here is a shift in relationships that we see. So normally in baseline years in April through June, the water quality and sport community dynamics has the lowest partial correlation when compared to the other relationships of latent variables. However, it's the highest in that post-kill period. The water quality and forage fish community dynamics still has the mid-partial correlation, but we really see a decoupling of the sport community and forage fish community dynamics compared to the baseline years where that partial correlation is relatively high. So in conclusion, constructed nested models through this framework can help create better fitting models and reveal that shift in relationship between the water quality sport and forage fish community dynamics and that shift we saw. these could potentially lead to fundamental changes in the ecosystem, especially if large events like what that happened in 2020 continue to occur and decouple these relationships. Ecosystems are complex, but SEMs are a potential transferable tool to assess changes in disturbances, especially if used consistently. The next steps for this project is to apply the same framework to the 2020 cyanobacteria bloom and compare the shifts that might have happened there after that event. Thank you to my funding sources, the Forest Risk Research Fellowship and Department of Biology, and all of the data sources in my lab. I don't know if I have time for questions, but my email is there just in case. Thank you.
So Dakota, nice job, and there is time for questions. And we have actually 2 questions coming in from Mitch, so it's a 2-for-1. After a fish kill, is there a change in species composition with one species becoming more dominant?
So after the fish kill, the black drum actually spiked in abundance, and they kind of drove the higher sportfish abundance. I'm trying to figure it out. So we did see a slight change, but there wasn't enough. So I ran a non-metric multi-dimensional analyses, and the true like composition or species that were there didn't change significantly. We still saw really similar species there overall as far as abundance.
And his second question was, have you looked at changes in species composition before 2011 and after? And I think everybody knows that 2011 is kind of that benchmark from when we started the blooms.
So for this specific project, I haven't. I was sticking in 2012, just mainly to kind of because I knew after 2011 things did shift. So starting in 2012 made the most sense for this project to get baseline years that were relatively more similar. But it's definitely something to look into, especially when going a little bit further.
As an angler, we seem to notice that this appears to be a greater abundance of catfish compared to trout, redfish, and snook. Do you see this in your data?
So the catfish, because I was looking more specifically at the sport and forage fish, they were definitely high, but I didn't consider them a sport fish in this sense because they're not often wanting to be targeted. And so when I was really looking at the trophic dynamics, they weren't in this specific assessment. But over time, the catfish do increase, but it's pretty consistent as far as what their numbers looked like while I was looking, because obviously I didn't look at the abundance of every single species in the system, but they are high relative to other sport fish abundances.
Okay, thanks, Dakota. Good talk. Next up, Andrew Kamarowski from Applied Ecology is going to talk about characteristics of wastewater contaminated groundwater in the Indian Lagoon. So you should be able to share your screen now, Andrew.
Okay, sharing screen. Okay, obligatory. Does everything sound and look good?
Yes, it does.
Okay, good morning. My name is Andrew Kamarowski, and I'm an environmental scientist with Applied Ecology. I'll be providing a brief overview of our ongoing project looking at groundwater contamination in Brevard County. This project began in 2017 with a pilot project funded through a Florida legislative grant through the Florida Institute of Technology, then continued with another grant through the Marine Resource Council, and then expanded county-wide through the Brevard County Save Our Indian River Lagoon program. The intent of this monitoring program is to overall describe the dynamics of groundwater nutrients in the lagoon area, allowing us to prioritize locations with higher groundwater contamination and also provide input information for pollution load models. And then additionally, interacting with homeowners is looking at behavior of septic tank owners, and then ultimately providing the baseline information needed to inform management decisions, and then evaluate the overall effectiveness of ongoing wastewater retrofit programs. So again, this project began in 2017 with Turkey Creek, shown in red in the south of the county, and then in 2018 expanded county-wide for 6 regions. starting with Titusville in the north, Merritt Island that's green, Merritt Island's blue, Suntree as green, and then Satellite Beach and Melbourne Beach, which will be referred to as the beaches for the rest of this presentation. In each of these locations, we have a septic community, sewer community, reuse, reclaimed, and natural. The septic communities, the well locations were guided by looking at using a swill model to identify where the plumes are expected to be to allow us to site as best we can the monitoring wells near those septic plumes. And overall, we have 45 monitoring wells throughout the county monitored monthly. And so we look at total nitrogen as composed of NOx, TKN, and ammonia, total phosphorus and orthophosphorus, which for this presentation we'll consider them the same. They're very highly correlated in our analysis. We also looked at fecal coliform, nitrate isotopes, and also soil organics. More information is provided about those items in our ongoing annual report, which I can provide the link to it if you're interested. So overall, for each of the treatment communities, there were 3 wells. And so I'll start first by looking at total nitrogen. And so graphed on the left is the median total nitrogen across all regions by treatment. Blue is reuse, red is septic, purple is sewer, and green is our natural control. As you can see, reuse and septic are by and far on average throughout the entire study period elevated in comparison to the natural control and the sewer. But even sewer is elevated over the natural control. The natural control locations are undeveloped or otherwise just vegetated areas such as the Turkey Creek Sanctuary. But as you can see, there's a lot more variability with reuse over septic. And now we break it down looking specifically at what makes up this nitrogen species composition. Septic was dominated by ammonia across the sample period, whereas reuse was dominated by nitrate, nitrite. So now if we pivot looking to total phosphorus, sorry, I had this 2 in there. If we look at total phosphorus, we can definitely see septic is by and large likely the most strong contributor to the total phosphorus concentrations in the groundwater. Compared to the natural, reuse, and sewer, they're pretty much about the same low, essentially background levels. But one of the other things we did notice is there does appear to be an increasing trend that is something we'll be looking into with this year's annual report. But when we break it down by year and the season, we also do see this trend generally play out where starting in 2020 dry season, we definitely see an increase in our extreme values and the overall range of values that we saw for all septic locations in the county for total phosphorus. So again, when we look at these by region, Turkey Creek and Suntree have Turkey Creek is generally high with Suntree recently increasing, whereas the beaches and Merritt Island are staying relatively low on the median total phosphorus, but there's still a fair amount of variability month to month within the wells. So Merritt Island shown in blue, the beaches in purple, Suntree in green, and Turkey Creek in red. And so when we break these down again by year and season, we notice that within Suntree, we see a drastic increase in the median value in addition to the overall range that we're observing post 2020 dry or after 2020 dry season. And then we see a similar thing play out in the beaches where the medians are still relatively low, but we do have more variability within certain wells. And then last, when we look at Turkey Creek, it seems to be a little bit more inconclusive, but it does appear that post 2020 we do still see a relative increase in the extreme values and upward movement in the range. So overall, for total nitrogen, we definitely see that with reuse and septic, there are elevated levels compared to the other landscape types. And the most key observation appears to be that the septic systems typically have higher ammonia, whereas reuse systems will have higher NOx. However, when we look at phosphorus, septic are definitely appearing to drive the majority of the total phosphorus variations. Our potential increases could be with something we're going to examine is that the post-use seasons in the post-COVID period may be a source or some influence on it. We have a number of other wells in other programs that we're looking at as well. They include business or business locations as well, wells. And then ultimately, we did see a lot of variability between our wells and other locations. And so this is going to be further research and examination through advanced statistical analysis such as PCA, and also looking through Kruskal-Wallis statistics in our annual report, which will be coming out later this year. So I'd like to thank our funders and partners at FDEP, Florida Tech, Brevard County, and Marine Resources Council. And I guess if there's any questions, I could try to address some of those.
Okay, thanks so much, Andrew. I don't see any questions that have come in, so what I'm going to do is encourage everybody for this and the other talks, go ahead and put your questions in, and the speakers will can continue to answer them in the chat and share them. So, yeah. Our next speaker, which is a student presentation, is Michelle Edwards, and she's here at Harbor Branch, and she's going to talk about the bull shark as a sentinel species for harmful algal bloom toxins. So Michelle, I see you're already on it. You have your content coming up.
Hi everyone, my name is Michelle Edwards and I'm a master's student here at Harbor Branch with Matt Ajamian. And today I want to talk to you about how we be able to bull sharks to better understand harmful algal bloom toxins in the Indian River Lagoon. So as we all know, harmful algal blooms have increased in the IRL in recent years. And this is an issue because many HAB species are capable of producing toxins, which may be dangerous to human and animal health. Unfortunately, the presence of toxins within the ecosystem is difficult to assess for a few reasons. First, the presence of toxic species does not necessarily indicate the presence of toxins. Second, there's a nonlinear relationship between algal cell density and toxin production. So a larger bloom does not necessarily indicate a greater amount of toxins present. And last, many toxins are known to persist in the environment, so toxins may remain in the environment despite the absence of a bloom. One way to estimate toxin exposure in an ecosystem that kind of mitigates these problems is the use of sentinel or indicator species that can act as kind of a canary in the coal mine and recognize the problems in the environment before they become a bigger issue. And elasmobranchs or sharks and rays are known to be useful indicator species because they are K-selected, so long-lived and slow-growing. They share food and water resources with humans. And the high trophic position of many shark species can provide kind of an integrative picture of a food web, alerting us to toxins in lower trophic species. The bull shark is the most common shark found in the IRL, and the IRL is in fact known to be a bull shark nursery where the species spends the first 9 years of their lives. Bull sharks are also known to be apex piscivores in the system, consuming mostly mullet, marine catfishes, and Atlantic stingrays. So understanding toxin present in bull shark tissues can also tell us something about toxins in these prey items. So the main objective of my master's thesis is to quantify baseline concentrations of HAB toxins in the tissues of bull sharks. And I'll be looking at toxin presence and toxin concentration and how they vary by tissue type, season, and region. To do this, we collected samples from 3 sampling regions across the lagoon: north in yellow, central in blue, and south in green. We sampled twice a year using longlines and gillnets in fall and spring between 2018 and 2020. We collected samples from 50 individuals for a total of 123 samples from 32 females and 18 males that ranged in size from 66 centimeters to 126 centimeters fork length. And so far, we've conducted toxin analysis on all but 11 samples. So today I'll be talking about 45 liver samples, 43 plasma samples, and 24 stomach content samples, which we screened for 14 different HAB toxins using UP, LC, MSMS, And then I ran statistics, including logistic regression and a series of ANOVAs to understand presence and concentrations and how they vary across space and time. So toxin screens have indicated the presence of multiple HAB toxins in the tissues of bull sharks sampled across the lagoon, including several variants of microcystins, nodularin, lingbiatoxin, domoic acid, okadaic acid, and brevetoxin. Each circle on these maps represents an individual animal, and the colors denote the type of toxin found in each tissue type, with liver on the left, plasma in the middle, and stomach contents on the right. What we can see is that animals that tested positive for one toxin often tested positive for multiple toxins. All of the detected toxins were detected across the lagoon, except nodularin in light blue and brevetoxin in red, which were only detected in a few samples and were spatially limited. Domoic acid, shown in yellow, and microcystins in green were the most common toxin types found. They were detected in samples collected from all 3 sampling regions, and logistic regression showed that the likelihood of detecting these toxins was not significantly different by region. All of the detected toxins were present in samples from both fall and spring sampling seasons, except brevetoxin, which again was only detected in a few samples. Again, looking only at domoic acid in yellow and microcystins in green, which were our most prevalent toxins, the likelihood of detecting each of these toxins was not significantly different between the fall and spring sampling seasons for any of the 3 tissue types. To put this information into perspective, we are seeing toxins across the lagoon, but in a relatively low percentage of samples. Domoic acid was found in 36% of liver samples, and microcystins were found in 42% of stomach content samples. But each of these toxins were found in a lower percentage of samples in other tissue types, and all other detected toxins, were found in less than a quarter of the samples that we tested. So these are box plots with the first and third quartiles indicated by the upper and lower boxes. The error bars show variation of the data. The median is the middle line and the X is the mean. The Y-axis here is toxin concentration in nanograms per gram, with domoic acid on the left and microcystins on the right. And to just further put this information into context, the highest concentrations we're seeing of domoic acid are under 250 nanograms per gram. The FDA actually considers 20,000 nanograms of gram of seafood flesh unsafe. So these concentrations are much lower than what is considered safe. The FDA has not established safe consumption limits for microcystins, but some states have, and that is 1,000 nanograms per gram. And the highest concentrations of microcystins that we are seeing are under 100 nanograms per gram. So although we are seeing higher prevalence of these toxins, they are in relatively low concentrations. The highest concentrations of both microcystins and domoic acid, and in fact, almost every other toxin we detected, were highest in the stomach content samples, which suggests that sharks are ingesting these toxins from their prey items. It also suggests that some bony fish in the IRL contain HAB toxins. So I ran some statistics on the data to determine if the stomach content concentrations were significantly higher than other tissue types. For domoic acid, a Kruskal-Wallis test suggested that concentrations were significantly higher, and a Dunn's post hoc test revealed that this difference was only in regard to plasma, but not liver. For microcystins, a one-way ANOVA suggested a significant difference for stomach content concentrations in relation to the other tissue types, but a Tukey post hoc test revealed the difference was only significant with regard to liver. This may be related to differing toxin characteristics and retention times, so more work is needed to understand these patterns. I also wanted to understand if toxin concentrations varied across space and time. Unfortunately, because few samples tested positive for toxins, low sample sizes prevented running statistical tests on the concentration differences. Obviously, this is good that we're seeing a lower prevalence of toxins, but it did prevent further analysis. However, I was able to look at domoic acid concentrations in liver. samples to assess differences in region and season, and a two-way ANOVA showed a significant seasonal difference in toxin concentrations, with spring concentrations significantly higher than fall concentrations. Pseudonychia, the diatom genera that produces domoic acid, is widespread in the IRL, with blooms occurring throughout most years since 2005. Domoic acid is not known to accumulate in the river because it's water soluble, but it may be passed through the liver during excretion from the body. So the DA present in the liver is likely to be from relatively recent exposure of the sharks. So this information suggests potential for seasonally elevated blooms and or toxin production in the IRL. Fusion multi-hab toxins were present in samples collected across the IRL, but they were in low prevalence and in relatively low concentrations. Domoic acid and microcystins were the most prevalent toxins detected, with the highest concentrations seen in the stomach content samples, and domoic acid concentrations in liver varied seasonally. Going forward, I plan to explore other variables that could have affected toxin presence or concentrations. In addition, understanding how bull sharks use the lagoon will help us to better understand the toxin data. And with that, I'd like to thank everyone that helped, and here are my references. And I don't have time for questions, but I could be wrong.
Okay, thanks, Michelle. You are correct on that, but you have an incredible number of questions in the chat box. Some of them are similar, but there's really a whole range of questions on your talk. So if you can, go ahead and answer them. And I think a lot of other people will be interested in those answers as well. So if you want to take your time doing that, that'd be great.
Sounds great. Thank you.
Thank you. Okay, our next speaker is Casey Craig from UCF. How efficient are oysters at excreting microplastics? And we can see your screen. It looks great. Okay, you were
Can you hear me?
Yes.
Okay, perfect.
Then you backed out though. You want to view the slides again? Go to slideshow. Now we're seeing the side panel and everything. So there you go.
Okay, so you can see my presentation and you can hear me.
Yes.
Okay, perfect. Yeah, so I'm going to be presenting some of my thesis research with you, which I touched on at the last symposium, but we're going much more in depth this time. So it's best to start by understanding how we classify plastics in environmental research, which is largely by size. So nanoplastics are the smallest. These are completely invisible to the naked eye and typically less than 100 nanometers in size. Microplastics, conversely, are a little bit larger than that. You can see some microplastics with the naked eye, and we define microplastics as any plastic object that is less than or equal to 5 millimeters in size. And microplastics are really what we're focusing on for this study. Macroplastics then would be any plastic object that's larger than this 5 millimeter size. But when we look at the microplastics group, we can kind of further classify them into these two subcategories, which are primary and secondary. Primary microplastics are basically plastic objects created at this small, less than 5 millimeter size. And these are things like nurdles and microbeads. Secondary microplastics start off as a macroplastic, undergo UV chemical or mechanical weathering, which then weakens them and they fragment into these now smaller, less than 5 millimeter sizes. The two most common types of secondary microplastics that we see are fibers and fragments, which are pictured here. It's no secret that plastics are ubiquitous pollutant in the marine environment. And with this, we're gaining a lot of information and a lot of research is being done on plastic ingestion in the marine environment. And so plastic ingestion has been documented in species of all sizes and trophic levels of marine biota. And really the consensus among the studies of ingestion are that microplastic ingestion has negative impacts on the consumer. So I have two examples here at the opposite end of the size spectrum. The first was a study done by Cole et al. Looking at microplastic ingestion in zooplankton, which you can see a great image of here. These bright green bits are actually the microplastics. At the opposite end of the size spectrum, I have a local example from 2019 in which a whale was stranded in Everglades National Park. FWC responded, The whale unfortunately died, and a necropsy revealed that plastic ingestion was indeed the cause of death. And this is the image of the plastic object here. So this leads me to a question, what about our organisms in the IRL? Where is our research regarding microplastic ingestion within the organisms that we have in the IRL? So when you do a search, there was a study done by Waite et al. In 2018 that found high concentrations of microplastics in oysters, the eastern oyster specifically, Crustaceae virginica, from Mosquito Lagoon. It's important to note that the species is harvested for human consumption above a size of shell length of 3 inches or approximately 76 millimeters. So while this study did confirm ingestion in the species, there are still some questions that came up from it, which we then turned into our study questions for this project, which are, what about ingestion of microplastics in oysters? Can they ingest microplastics? And secondly, with what efficiency can the oysters ingest the microplastics? To answer this question, we partnered with Smithsonian Marine Station and used their flow-through bivalve filtration tanks, which you can see pictured here. What these machines really allow us to do are to place a live individual oyster into this chamber where we can control the flow rate of water into the chamber such that we can mimic the natural water conditions and they can resume their normal feeding behavior. Now, oysters have 2 methods of egestion or excreting, which is through pseudofeces and feces. Pseudofeces is particulate matter that is brought into the oyster tissue, is elected not to be digested, and it instead is encased in mucus and excreted through pseudofeces. Feces, conversely, would then be particulate matter that is brought into the oyster tissue, is elected to be digested, and is then excreted. So you can tell the difference between the feces and the pseudofeces is quite apparent in these 2 images here. Again, the pseudofeces is the loosely just mucus-bound material, and feces is actually digested. So we collected small and large individuals from the Indian River Lagoon, where we then placed them into these individual chambers. They acclimated for a period of time, and then over a 2-hour trial, we would independently collect their pseudofeces, feces, and at the end, collect the organism itself. From that point, samples were brought back to the lab where oysters underwent chemical digestion using a potassium hydroxide solution. They were then vacuum filtered to extract microplastics. The biodeposits were also vacuum filtered and then rinsed with ammonium formate to remove any excess salt. When we look at our results, particularly regarding just the ingestionability, we can see that both small and large oysters are capable of excreting microplastics through both their feces and their pseudofeces. And these are at significantly different amounts. In fact, oysters will excrete microplastics through their feces significantly more than in their pseudofeces, which is an indication that they are actually digesting more plastics as opposed to just excreting them before digestion. And when we look at the abundance of microplastics in the oyster tissue, we also see a significant difference from that which we see in the biodeposits. And this is all confirmed by a generalized linear model, When we look at the mean microplastic size, we also detect some differences. So while there isn't a difference between, a detectable difference between the feces and the pseudofeces in terms of the microplastic size that was excreted, there is, however, a pretty large discrepancy between the microplastics found in the oyster tissue, the size of the plastics found in the oyster tissue, versus the size of the plastics found in the biodeposits. In fact, in the oyster tissue, microplastics are on average 1.8 millimeters larger than those found in the biodeposits, which would signal that potentially these larger plastics are in fact getting trapped in the oyster tissue. When we normalize the rates of excretion, what we really get is these oysters are capable of excreting 1 microplastic per 1.5 hours through their feces and 1 microplastic every 2 hours through their pseudofeces. When we look at the ingestion efficiency data, we see a lot of variation. In fact, the mean ingestion efficiency is 53.5%, right smack dab, pretty close to the middle. So I just want to orient ourselves with this plot, which is the data, the ingestion efficiency data. On the x-axis, we have mass of the oyster in grams. On the y-axis, we have the proportion of the microplastics that were actually ingested. This red line right here is marking the largest spat that was classified in this study by mass, which is 3.7 grams. And in this black box, we're looking at the 100% excretion data, or the oysters that were able to clear all of the plastics from their body tissue. And what we really see is just a clear drop-off in egestion efficiency as the mass of the oyster increases. And this was also confirmed by a GLM, which determined that mass is in fact the best predictor of egestion efficiency. So our smaller oysters are more efficient at egesting microplastics than our larger oysters are. We also elected to do FTIR spectroscopy on a subset of our samples found, or a subset of the microplastics found in samples. And PET was determined to be the most abundant polymer found in both the oyster tissue and the biodeposits. So PET is polyethylene terephthalate, and it comprised almost 70% of our samples, which you can see in the pie chart on the right-hand side. Other types of polymers that we found were polypropylene and polystyrene, and we did find one instance of a polymer blend. So the takeaways from the study really are that oysters, the eastern oyster, can ingest more microplastics through their feces than through their pseudofeces, again indicating that they are actually digesting this material. The Indian River Lagoon oysters contain a mean of 1 microplastic per gram of tissue weight, which is also 1.7 microplastics per individual, which is much lower than the concentrations that were seen in the Mosquito Lagoon study prior. As I mentioned, the smaller oysters are more efficient at egesting microplastics, which would really make sense because a larger oyster will have been in the environment and exposed to more plastics than a smaller oyster has. And again, PET was the most abundant polymer that we found in both the oysters and the biodeposits. But this result is particularly interesting because when we look at PET, or when we look at polymer production by mass, PET isn't even in the top 3. In fact, it's not in the top 5 most produced polymers by mass. It's listed as number 6. So this begs the question of why are we seeing such large abundances, proportions of PET in the environment? And so when you do some basic research, you can find an answer pretty quickly. So I have this blurb here, which is straight from the PET Resin Association. PET is a clear, strong, and lightweight plastic that is widely used for packaging foods and beverages, especially convenient-sized soft drinks, juices, and water. Virtually all single-serving and 2-liter bottles of carbonated soft drinks and water sold in the United States are made from PET. So essentially, PET is correlated with single-use plastics. Look for the number 1 triangle on all of your items, and that'll tell you that it's PET. With that, I would just like to acknowledge our funding source, the IRL NEP, and of course, my wonderful collaborators who we had so many working on this project. And with that, I can go ahead and take any questions if there's time, or we can move on.
Yeah, hi, Casey. I think that was good. I don't see any questions that came in right away, but I suspect you'll get some now. And it looks like answering questions in the chat has worked out really well, and people like to see those answers. So you guys can still send questions in and Casey can reply. So next up is our last speaker of the session, who also is a student, Rachel Stark from FIT. And Rachel will talk about changes in the abundance and distribution of benthic mollusks and sediments. So Rachel, you should be able to share your screen. It says it's starting to.
Oh.
It's coming up.
Okay. Can everybody share the screen?
Yep.
Okay.
You just need to go to from beginning up top there. Where
Yeah. Okay. All right.
There you go. Looks good.
Thanks. Hello, everyone. My name is Rachel Stark. I'm a master's student at the Florida Institute of Technology, and I'm going to talk to you about my ongoing study concerning the changes in the abundance and distribution of benthic mollusks and polluted sediments of a shallow subtropical estuary. So environmental dredging is a remedial process that has been carried out in several locations along the Indian River Lagoon in an attempt to remove contaminated sediments that are high in fine-grained organic rich sediments. This is an attempt to improve the water quality and restore the benthic habitats along the Indian River Lagoon. Due to several decades of nutrient pollution, the Indian River Lagoon benthos has accumulated many of these fine-grained organic rich sediments that are colloquially known as muck. IRL muck can be defined by comprising of 10 to 30% organic matter. 60% silt and clay, and a water content of about 75% by weight. And this is estimated to cover around 10% of the Indian River Lagoon, ranging from a few centimeters to several meters in depth. And muck exacerbates the IRL's eutrophication and habitat degradation issues through water column resuspension, harmful algal bloom fueling nutrient fluxes, benthic light shading, and hypoxia induction, and overall benthic smothering. I chose to use mollusks as bioindicators of the effectiveness of the environmental dredging as a restorative process. They are second only to arthropods in overall taxonomic diversity and are usually very abundant. Many of them are sensitive to environmental changes, but many of them also exhibit wider ranges of tolerances for living in polluted sediments. They are also relatively sessile overall and lead long cycles than many other beds. They are also arguably a little more easily identified to lower taxonomic levels than other members of the benthic community. Muck can have many damaging effects on mollusks. Their fine gills and other structures can get clogged by silt and clay particulate, which could end up suffocating them. They also are subject to trophic amensalism, where deposit feeders competitively exclude filter feeders by disturbing and resuspending these sediments, which end up clogging their filter feeding mechanisms. And since muck is so easily resuspended, smothering and suffocation are significant threats to benthic mollusks living in these degraded habitats. And this can also lead to habitat loss through benthic smothering, overall resuspension, blocking light, algal bloom fueling nutrient fluxes, and so on, leading to overall habitat degradation as well. So I hypothesize that there is an inverse relationship in the community composition of mollusks with percent sediment organic content. And by removing these sediments with high concentrations of fine grain organic rich matter, this will increase and benefit the mollusk benthic community as a whole. So we went out sampling quarterly from March 2017 to October 2020 in the MIMS region of the IRL. The MIMS boat ramp was dredged out in the summer of 2018, which made it an ideal location for the study. In the map on the bottom, you can see all of our sites. The red dots indicate areas where muck was dredged out. The green dots indicate seagrass sites that are proxy to but not in the dredge line. And the orange dots will indicate control muck pit sites, and the blue dots indicate control seagrass sites that were also not dredged. The control site was chosen as it was the closest area to this dredged area that also had seagrass. So we, like I said, we collect this data quarterly. We used a petite ponar grab to get benthic samples, 3 of which were sieved in a 0.5 millimeter sieve in estuarine water. One of the grabs was left unsieved, and that was used for sediment characterization purposes. The 3 grabs that were sieved were used for in-fauna analysis. So we took those samples back to the lab and either left the samples whole if they were able to be processed in a appropriate amount of time. Otherwise, we had to cut them just for time constraint purposes. We split this 1 to 4 times. We used light stereomicroscopy from 8 to 35 times magnification to count and identify any apparently alive at collection organisms and identify them to the lowest taxonomic level. For the sediment characterization, we used an analysis for silt clay, water content, and overall percent organic content. The silt clay content analysis was conducted by taking 10 grams of samples that had visibly higher muck content, and any samples that were sandier, we used 30 grams for accuracy. They were sieved through a 63 micron mesh to separate silt and clay particulate from larger sediment particles. And then these samples were baked at 105 degrees Celsius for 24 hours and then reweighed, which gave us the overall silt clay content. For water content, we used a very similar process where we weighed unsieved sediments prior to and after baking them at 105 degrees Celsius for 24 hours. For the organic content analysis, we used the mass loss on ignition method, where the sediment was dried, ground, and weighed before and after baking at 550 degrees Celsius for 4 hours. Now, just to reiterate, this is still an ongoing study, so I'm going to show you some preliminary results that I have right now. This is just kind of like a preview. I made this overall parenthesis absence analysis where I looked at every single organism that we identified at each individual site over the course of the 4 years of the study and tried to figure out which ones might have had the biggest impact via dredging. The red stars will indicate those that we found to be present only pre-dredging, and the green stars indicate any organisms that were present only post-dredging. So Amygdalin papyrium and Haminoia cicinnea were found to be only present pre-dredging at the treatment site, and Japanactyon punctostriatus was found to be only present at post-dredging at the treatment site. So this could indicate that dredging was beneficial for Japanactyon punctostriatus. Again, analyses are still being conducted. So just to kind of orient ourselves with this graph, I did a generalized linear model and found that mollusk abundance with sediment percent organic content was found to be statistically significantly correlated. So I created this graph for that. The y-axis is the abundances per meter squared, and the x-axis represents sediment percent organic content. The graphs on the left and the right are actually the exact same data. I just zoomed in kind of on this area that's highlighted in the red box so that we can kind of get a better idea of what's going on over there. And as you can see, there does seem to be an inverse relationship with mollusk abundances and sediment percent organic content, which does support my first hypothesis. I would like to thank the Brevard County Natural Resources Department and the citizens of Brevard County for funding this project with the Save Our Indian River Lagoon half-cent sales tax. And I'd also like to thank the Florida Institute of Technology for providing me with all the resources needed to be able to conduct the study. And I'd also like to thank the members of the Johnson Lab, past and present, for helping me out with this study.
Well, thank you. That was a good presentation. I don't see anything in the chat, but keep an eye on it.
Okay.
We're not going to anything down in the meeting, and please share it with everybody. Seems like people like to see the answers, just like we would if we were all together. So we're now segueing to a 3-minute session. Kelly Hunsucker, who is also on the steering committee, will take us through a series of rapid fire presentations. And then after that, we'll take a short break.
Awesome. Thanks, Dennis. So I'm keeping a timer in case you guys don't know how much time you have left as well. But we have the 3-minute, 3 slides. So one of my favorite parts of the conference. And to kick us off for today's 3-minute section, we have Matthew from Harbor Branch.
All right. Hello, everyone. I am a master's student at Harbor Branch, and today I will be talking about my thesis research. This project has been a collaborative effort between researchers at Harbor Branch, the Bigelow Laboratory, and Florida A&M, with funding from the National Academies of Science, Engineering, and Medicine, and the Harbor Branch Oceanographic Institute Foundation. The goal is to determine how redox conditions affect the degradation of harmful organic contaminants in marine sediment after an oil spill. Following such an event, through marine oil snow formation and other physical and chemical factors, toxic hydrocarbon compounds are deposited on the seafloor where they can persist for years. On the seafloor, these compounds can be degraded through microbial respiration and other abiotic redox reactions. Under anaerobic conditions, microbes will respire using a range of different electron acceptors, including iron, and in particular, iron-reducing bacteria efficiently degrade hydrocarbons, and as such, are commonly employed when removing organics from contaminated soils. One particular iron-reducing species, Schoenella onadensis, has demonstrated exceptional ability to accelerate the degradation of hydrocarbons by catalyzing the Fenton reaction shown on the bottom right, which both generates reactive hydroxyl radicals capable of oxidizing hydrocarbon compounds and recycles the reduced iron. We use this species of bacteria as well as mineralogical amendments to see if iron reduction could have the same effect on estuarine sediment. Next. To test this, we collected sediment from the St. Lucie estuary, which was then mixed with either quartz sand, iron-coated sand, or iron-coated wood chips developed by Dr. Vivian Merck at FAU and placed into cores. Some cores had Schoenella culture added as well. Crude oil was mixed into the top centimeter of these cores, which we then incubated for 9 weeks in the Harbor Branch Sediment Incubation Laboratory, which I designed and built specifically for this project. This lab has a flow-through system that filters and chills water from the Harbor Branch Channel, which then flows into the individual tanks holding the sediment cores. And replicate cores were pulled at 4 time points for hydrocarbon, iron, and microbial DNA analysis, allowing us to follow the evolution of the sediment throughout the experiment. Next. By analyzing the pore waters from the different cores after 3 weeks, we determined that the addition of iron-coated sand significantly increased the concentration of dissolved iron 2 and iron 3, meaning microbial iron reduction was induced. We also saw that the addition of Shonella to the cores with iron sand effectively doubled the concentration of dissolved iron species, confirming that this bacteria did increase iron reduction. While we are still awaiting hydrocarbon data from the final time point, results thus far, shown on the right side of this slide, suggest that the amendment of natural sediment with iron accelerates the initial degradation of both polycyclic aromatic hydrocarbons and alkanes found in crude oil, and that the presence of Shonella onadensis has further accelerated the degradation of these compounds. And that is all. Thank you.
Perfect timing. You just beat my timer. So next we have Ash, also from Harbor Branch.
So I'm a postdoc at FAU. I'm an ocean modeler, and I'm going to be talking about the residence times of water inside the Indian River Lagoon. So how we do this is we set up an ocean model around the lagoon. So this is a ROMS ocean model here, and this is our domain. We do some validation on our ocean model. So here we see some observations and model comparisons, so we can see everything is kind of lining up on this line, so we can see our model is doing a reasonable job. And we look at the residence times of the water. So how we define this is we have a tracer that is placed just in the lagoon, and we measure the time it takes for that tracer to become half of the concentration, or more or less. So what we see here is that the northern part of the lagoon has a very high residence time, so this water is kind of stagnant. So this is a kind of known fact already. However, the dynamics that control that process has not been explored yet. So here we kind of look at some of the processes that might affect the water residence time. So what we see is the effect of the Gulf Stream. So we can see the Gulf Stream kind of affecting here around the bottom. And what that does is it pushes water into the lagoon, and we see the water getting pushed right into the northern part of the lagoon. And what that does is it creates a sea level difference between the lagoon water and the shelf water, which will drive some sort of flow. The second process is the wind. So the wind is kind of highly correlated to the sea level in the lagoon. And the effect that the wind has is it comes into the north part of the lagoon, kind of drives a flow down into the northern part of the lagoon, and that stagnant water that was there before is then being pushed out of the lagoon. So that's another process. And finally, we do one more experiment where we look at the impacts of the river flow. So we have a control flow, we have a control simulation, and then we have another simulation which has no rivers included. We take those two experiments away from each other and we can see the effect. So what is happening here is we have fresh water entering the northern part of the lagoon, and that is driving a flow outside of that northern part. Okay, and that's me. Thanks very much.
Perfect. Thank you. So let's see. You were right on time. And the next presenter we have is Angelo from UCF.
Hello, everybody. Okay. Okay, good afternoon. I am Angelo Hernandez, UCF student developing a 3D hydrodynamic model for the Mosquito Lagoon. By encasting deep time conditions and analyzing current ones, we will recognize trends that will allow us to relate significant changes of the habitat and ecology to these possible variations of the hydrodynamics through time. We will model the accurate flow paths through the northern portion of the Mosquito Lagoon with high complexity due to the morphology of the lagoon, such as sand barriers and mangrove islands. Also, we will be predicting future conditions to encounter how managed inlets will affect flows, temperature, habitat, velocities, and ecology. We will show relevant information for future decision making. Next. Now, the objective of this research is to use present-day hydrodynamics to model future and deep time changes in the Mosquito Lagoon. As previously mentioned, how historical changes could affect the ecology and habitat. This will help other scientists to confirm their studies. An example of this is Jacqueline Rumberger from Washington State University, which has talked yesterday about the multidisciplinary research on reconstructing the IRL over deep time. Furthermore, we will help to the decision making by investigating the management of inlets. The method used is the 3D flexible mesh modeling software, which allows us to do hydrodynamic simulation, especially in this study that is wind and tide diving. This software uses the shallow water equations as well with the sediment transport equations for computation in each mesh. Next. Now, the work in progress. First, we collect the data necessary for the present day conditions, such as wind, water level, and humidity data from different sites, such as USGS, NOAA, and NASA Giovanni, using the best spatial and temporal resolution. Second, the computational mesh was created by taking special attention to the delineation of the morphology, again with the best spatial resolution possible, choosing main channel as flow path from the northern portion to the rest of the lagoon. As you can see in the bottom right figure, there is the correct delineation of from the Ponce de Leon inlet to the rest of the lagoon for two main channels. One of them is the intracoastal waterway, and we're trying to take into account other channels that can affect the hydrodynamic of this lagoon. Then ArcMap was used to join the grid along the bed level data. And to conclude, the model validation will be conducted by comparing the output of the model with actual water level and velocity data available in our lab and different sites previously mentioned. Thank you.
Very nice, right on time. So next we have Gabriella from UCF as well.
Good morning, my name is Gabriela Ford, and I'm going to present a field experiment carried out, the hydrodynamic impact of boat wakes to shoreline ecotones. The degradation of estuarine shorelines has been an ongoing problem resulting from coastal development and anthropogenic alteration. This is a worrying subject because shoreline erosion degrades coastal habitats and lessens nutrient cycling. Associated to this, boat wakes have demonstrated to produce larger in magnitude waves than wind, as you can see in the video. In high traffic areas, these large magnitude wakes could also occur frequently enough to change sediment transport patterns and cause erosion. Also, in previous studies, it was demonstrated that even small recursion of vessels from 100 meters away could also cause a negative impact in the shoreline. With this, the objective of this study was to observe the hydrodynamic signatures of boat wakes generated from varying distances from shore and to characterize the potential effects of boat wakes on sediment displacement from an eroded shoreline in the Mosquito Lagoon seen in the figure below. Next slide. Figure 2 displays the study site located in Mosquito Lagoon. And the insert diagram above shows the experimental setup where 4 to 12 replicate boat passes at 50, 100, 150, and 200 meters away from shore, where wind speed, velocity, and water surface deformation conditions were recorded, from which significant wave heights, energy to shoreline, and orbital velocities were calculated, seen in figure 3. Data taken during boat wakes were characterized by the green shading seen in the figure and the are the unshaded data between the boat passes. Additionally, core samples of shoreline sediments were taken to calculate critical shear stress and to determine if shear stress produced by boat wakes can transform sediments, which could potentially promote erosion. Next slide. From the results from the experiment as seen in figure 4, the boat wake signatures could be distinguished from ambient conditions at all distances. Significant wave heights, the top graph, were seen for each boat pass were much higher much higher than ambient waves, and were very similar throughout distances, and even a little higher from 200 meters from shore, which was not expected. We were not even certain we could detect signals from 150 meters beyond, and it was also expected that significant wave heights would reduce as we were further from shore. Additionally, it was surprising to find that cumulative energy increases considerably as distance increases from shore, as well as shear stress as we go further from shore. The increase in energy with distance is because wave heights last longer than the ones generated from closer to shore. From figure 3 and 4, shear stress produced by ambient conditions delineated by the lower dashed line, we can see that these were competent to move smaller grain sizes up to D40 compared to the shear stress produced by boat wakes that showed to be likely competent to move bigger sediments up to the medium grain sizes, indicating that the boat wakes we observed were likely to be erosive. Thank you.
Nice. Thank you. It looked like your video worked a little bit right at the end there. We saw a little bit of it. Next up, we have Lorelei from Florida Oceanographic Society.
All right. Thank you for the introduction, Kelly. And just a quick special thanks to my collaborators at Martin County and FWC. Next. We spent a lot of time during the pandemic here in the Jensen Beach impoundment, which I think is going to pop up in a second.
Next slide there, Brian.
Perfect. Thank you. So this is the Jensen Beach impoundment. It contains 61 hectares of mangrove swamp. And just to orient you really quickly here on the east side, this is where you see A1A. Down to the south, this is Northeast Causeway Boulevard. That takes you over the Jensen Beach Causeway into Jensen Beach. And so this is on South Hutchinson Island. And this right here is an active mosquito impoundment. And the blue dots that you see, they represent a series of culverts and pumps that facilitates the exchange of water between the north and the south cell, as well as the cells in the Indian River Lagoon. Next. Unfortunately, in the aftermath of Hurricane Irma in 2017, there was a mass, back one more. Thank you. Back in 2017, Hurricane Irma, mass mortality in the southern cell, which killed off about 22 hectares of mangroves, and this was due to high water levels and poor water quality. And in an attempt to try to restore these mangroves in the southern cell, 4 culverts have been added. Well, actually 3 have been added. 1 more is on the way, but this was done in 2020. And the idea was to just increase connectivity between the north and the south cell, as well as the south cell to the IRL. And so this set up a wonderful experimental design for us, with the main question being, what is the trajectory of ecosystem service recovery after hydrological restoration? So what we did was, before these culverts went in, we set up 3 transects in both the north and the south cell. Up in the north, you can see the red, that's the transects, and along each transect, there's 3 permanent plots. Down in the south, you can see the yellow, and these permanent plots are essentially the mangrove mortality, which we're going to use to compare to the healthy mangrove populations in the north. And so we're going to essentially use space for time substitution to better understand ecosystem recovery after the changing in hydrological conditions. So next. So without getting into all the details, this is what one of the south cell plots looks like. And there's many different aspects of ecosystem recovery that we're interested in, whether it's elevation change, sediment dynamics, water quality, soil efflux. I personally am interested in carbon storage. And I'm sure many of you here in the audience know that mangroves can sequester huge amounts of carbon in their biomass and store it for millennia in their accreting sediments. Next. So just to jump straight to the results, that die-off of the 22 hectares actually resulted in the transformation and loss of about 1,700 megagrams of carbon from above ground biomass. And this is equivalent to the carbon dioxide emissions from about 723 homes' energy use for just 1 year. Next. But there is a silver lining. Every 6 months we go out to sample, and so far that natural recruitment into the system has restored about 267 megagrams of carbon of that 1,700 that were lost. And so we anticipate an increase in carbon with every sampling period that we go out. So please stay tuned for more work on the return of ecosystem services from this crew in the Jensen Beach Impoundment. Thank you.
Great. Thank you. Next up, we have Morgan from Florida Tech.
Awesome. So today I'm going to be talking to you about the Living Docks program here at Florida Tech. So the Living Docks program is a citizen science initiative that aims to improve water quality through the introduction of oyster restoration mats. Founded in 2013 by Dr. Kelly Hunsucker and Robert Weaver, the project utilizes dock pilings. Oyster mats are constructed out of UV-resistant plastic with 60 to 80 dead and dried oyster shells attached and are then wrapped around these pilings. Some benefits to using the oyster mats include the lack of a permit or no permit because it's not on the seafloor. They're easy to assemble and can be applied anywhere. They encourage a healthy, diverse community of organisms through a natural substrate like the oyster shells, which we obtain from local restaurants in the area. The project is primarily driven by community interest and participation. So far there are 12 locations stretching from Cape Canaveral to Sebastian. For my research, I analyzed 9 of the 12, and I looked at temporal and spatial variations amongst the locations. Next, please. Right. So the shells I chose were randomly selected and denoted by colored zip tie, which you can see on one of the shells here. On the left, we have our Cape Canaveral location, which is our northernmost location. And on the right, we have our Sebastian location, which is our, Sebastian is the southern and Cape Canaveral is the most northern. The top 2 photos are from our November assessment, which was the warmer temperature. And the bottom 2 photos are from our January assessment, which was the cooler water assessment. So you can see for Cape Canaveral, there was an influx in cresting bryozoan, even covering the mesh itself. And for November, there really was an influx in arborescent bryozoan and algae and oysters as well. We lost a few of our samples. Next. I also decided to do a filtration experiment. So I ran a 40-hour filtration experiment looking at total suspended solids and chlorophyll A. 4 out of the 9 docks were chosen, with 2 of them being Cape Canaveral and Sebastian. Samples were collected and then vacuum filtered at 0 and 48 hours. And if we look at the graphs on the right here, location is at the bottom on the x-axis, and the y is the total suspended solids, milligrams per liter. Sebastian had an increase for the November and a decrease for the January samples, and then IAP decreased both times. Lighthouse Cove unfortunately increased, possibly due to a fungus that grew in the shells, and A1A had a high amount of oysters, so I think that is due to the immense decrease that we saw there. The increase in Sebastian in November could be due to the influx of silt on the shells as well. So ultimately, I still have to look at my chlorophyll A samples and assess suspended solids and organism presence and abundance. Thank you.
Nice, thanks, Morgan. Next we have Olivia from the Brevard Zoo.
Good morning, everyone. So today I'm excited to talk about a project that the Restore Our Shorns team performed with high school students here in Brevard County to restore seagrass last summer. So you can see 2 of our happy, excited teachers, part of our Grasses and Classes program. So we had 2 goals with this project, and the first goal was to engage high school students in every aspect or each aspect of seagrass restoration hands-on. And then the second goal was to see if simple by hand planting methods of seagrass fragments could lead to successful restoration in our part of the lagoon. So in early March, we set up closed tank systems in 2 high school classrooms. using shoalgrass fragments from FOS's seagrass nursery. We planned to grow those out in the classrooms and have the students care for them and then plant them in late spring. But unfortunately, next, schools ended up closing about a week or two later for spring break, and they did not come back until August. So we had to change our plans a little bit. Next. We decided to push back our seagrass planting until late May, and we used only staff for the planting. We planted about 48 meters squared of seagrass fragments, new fragments that we also got from FOS again. So we wanted to see if a biodegradable bamboo staple could help anchor fragments into the sediment. So half of the plots were planted with the staples, half without, and we also planted at 2 different depths, so shallower and a deeper depth. Since we lost our high school student volunteers, we decided to reach out to our zoo team volunteers here at the zoo, and we had an amazing response. So they helped us monitor the project for 4 months. They became expert seagrass monitors. Next. So how did the seagrass do? The graph on the left shows that the seagrass growth and shoot density peaked in July. So this was Haliduli right-eye shoalgrass. So that's consistent with what it would be a normal growing year. We did also see that growth and survivability dropped after week 8 as the water level rose, so probably less light available for the seagrass. On the right, you can see that we did not see much of a difference between the plots that had the bamboo staples and those that were just bare root planted. So in the future, we probably won't use the bamboo staples. Next. However, we did see that the deeper plots had a little bit better survival. So maybe the shallow plots had a lot of wave action influence that may have pulled out some of the fragments. Overall, growth and survival did drop as the water level rose, and we saw algae bloom start in August. So what did we learn? Adapt and keep going. There's always ways to keep a project going even during a pandemic. Maybe we should plant a little bit earlier, like April, so that we can maximize the growing season. There may also be a Goldilocks depth zone, somewhere that's not too shallow and not too deep. And then success is possible with simple methods. So we planted all this by hand, and it's possible to use in the future volunteers to perform a similar seagrass restoration project. Next. So thank you to all of our partners and our funding partner, the IRL NEP, EELS, all the high schools, and then of course, the rest of my team at the Brevard Zoo. Thank you.
Great, thanks Olivia. So next up we have Emily from the Indian River Lagoon Aquatic Preserves Program.
Hello everyone. Can you all hear me okay?
Yes, yeah, loud and clear.
Great. Okay. So my name is Emily Sermont. I'm the Shoreline Restoration Project Coordinator for Indian River Lagoon Aquatic Preserves, and I will be talking to you today about how we have been able to continue to engage our volunteers through vegetation husbandry during the COVID-19 pandemic. Next slide, please. Our plant fostering project can be thought of as a 2-part program consisting of educational exposure and hands-on service learning. Pre-COVID, we used vegetation husbandry mainly as a way to engage high school science classes. However, over the last few months, we have expanded our program to involve camps and after-school programs. So instead of giving in-person presentations as we have in the past, we put into action 2 safer alternatives. We have conducted virtual presentations, something that we are all familiar with, and we have created handouts, which can be given to participants in lieu of presentation. So virtual presentations, providing handouts, or in-person presentations with masks and social distancing are all ways to continue following CDC guidelines and continue outreach. We have found that Zoom presentations work best with high school age science classes in the classroom environment, where handouts have been really successful for camps and after school programs where participants come and go and attendance fluctuates. Next slide, please. So the vegetation husbandry aspect of the program is essentially the same pre-COVID and today, since plant care can easily be done in solitude. So for a designated period, participants ensure plants receive adequate water, sunlight, and they can experiment with propagation. And then the plants are collected by our office and used in shoreline restoration projects. Currently, all of our plant fostering volunteers are caring for Sesuvium portulacastrum. We chose this plant as our primary plant and really as our starter plant for its ease of care, as well as its capacity as a stabilizing ground cover. Supplies drop-off and pickup can easily be done following CDC guidelines or through completely contactless exchange. And participating teachers, camps, and after-school programs, they can determine the safest way to care for plants according to their own organizational guidelines. At our office, we have been lucky to use small groups of volunteers for outdoor planting events, utilizing masks and a plethora of hand sanitizer, but we realize this might not be an option for all other organizations at this point. Vegetation husbandry has been a lifesaver for the Shoreline Restoration Project, allowing us to expand our outreach during a pandemic and continue to allow opportunity for hands-on service learning. COVID-19 has challenged all of us in a variety of ways, but I hope this presentation is helpful for others who continue to look for creative activities to engage volunteers for the next couple months. So thank you to the Indian River Lagoon National Estuary Program for funding the Shoreline Restoration Project, and thank you all very much for watching.
Great, thank you. So our last presentation of the 3-minute section will be given by, is it Mandy and Carol from University of Florida? Or is it just Mandy?
Mandy and Carol.
Oh, perfect. Well, wonderful. Whenever you guys are ready.
All right. So we are excited to tell you about the work our civic team is doing to address awareness of water quality issues related to the Indian River Lagoon. The civic program consists of University of Florida and Florida A&M University Extension faculty working together to help communities address issues through facilitated discussions that seek to be inclusive, informative, and productive while providing science-based, non-biased information to help our audiences make better decisions for their lives and for the water quality. For almost 4 years, Civic has worked with groups across the state to explore issues related to conservation, sustainability, disaster preparedness, affordable housing, and natural resources protection. And our endeavor seeks to bring people together to deliberate and find common ground and shared solutions that lead to stronger communities. Next slide. Yeah. And Mandy was going to take it from here. I'm not seeing her, so we're using up our time. Let me see. Okay, so since Mandy's not available right now, to ensure that Civic's commitment to inclusive engagement is not mere lip service, work entails finding local leaders who are not typically seen at meetings nor heard in discussions, in this case around water quality of the lagoon. So inclusion is a relative term, and what it means to you depends on the part of the community you're a part of, your focus, your location, and even timing. Civic took inclusion in a region with a majority population that's white to mean actively connecting those who represent African American, Latino, and Latina, Asian, and other small minority groups. And this past October, we held a facilitated forum online, after which participants requested that Civic continue to hold meetings with an African American community in the city of Fort Pierce. And the intent was to explore what's known about the potential human health problems related to subsistence fishers and the long-term contact with the contaminated water. Subsequently, a collaboration town hall was held during which the local scientists shared her organization's research on fishing consumption, as well as her desire to form relationships in minority communities. The opportunity provided by these civic supported meetings catalyzed the integration of various networks with one goal. And then based on the discussions and feedback from our forums, we formed an action team that looks at water quality from a number of angles. And the team includes University of Florida, FAMU faculty and grad students, county staff, city officials, community change makers, and researchers studying the sources of pollution and the health effects of water quality on the least resource populations on the Treasure Coast. Going forward, Extension's role through Civic will be to engage more of the community around the issue, provide education, and help identify resources to work to bring identified solutions to fruition. Together, we can include all members of our community to make a better future. Thank you.
Thank you, Carol. It looks like Mandy was there. We just couldn't see or hear her.
Oh, I'm so sorry.
So thanks for finishing the presentation.
Technology is wonderful when it works.
We have a 10-minute break. So Dennis, what do you think, 11:45?
Yeah, actually, Kelly, we're going to go with a 5-minute break because we're behind. So that'll be, I think, we're like 11:41-ish. And I know I see Holly and she's getting geared up for the next session. So we'll do a quick 5-minute break. Thanks so much for your work here, Kelly.
All right, everyone, welcome back from your very short break. We're going to keep the ball rolling and kick off our very last 10-minute presentation session of this conference here. And this is going to be oral session number 4 on IRL biodiversity. And I'm very happy to be introducing our first speaker, Maria Cristina Diaz, and she's going to talk to us today about IRL sponge biodiversity. Take it away, Christina.
Thank you, Holly, and thank you everyone for being here. On 2020, a group of scientists from Harbor Branch, scientists and students from Harbor Branch Oceanographic Institute and Smithsonian Marine Station in Fort Pierce started a collaboration to deepen our understanding on sponge biodiversity at the Indian River Lagoon. Today, I will summarize our update results on species richness and taxonomic diversity. Before we started, we knew there were 16 species reported in peer-reviewed publications that range from 1991 to 2018 and were mainly related to ecological studies in the system. And 51 morphospecies were listed in IRL species inventory that are considered great literature. We suspected that considering habitat diversity and the widespread latitudinal range from subtropical to temperate zones, that our vision of sponge biodiversity was an underestimation. So we embarked to explore from Jupiter Inlet in the south to Mosquito Lagoon in the north between June and September 2020. And we used an integrative approach where when we included external morphology, skeletal element studies, ultrastructure or skeletal architecture when necessary, and a molecular approach that will be presented to you right after me. Our goal was to have the best species characterization that would allow us to identify and maybe draw some phylogenetic relationships. What we have found after processing 2/3 of our 261 samples is that we have gone from 51 morphospecies to 81, which means an increase of 60% on species richness. And we know that we are more to come. We have added one order, the Chondrocida, and 3 families. And as you can see in the graph below, not all orders are represented equally. There are 5 orders that have between 8 and 19 species, and they're the most specious. And you can see that they are all in artificial habitats, but some taxa within these orders are in mangrove habitats or seagrass or oyster reef. I could mention these orders include Haplosclerida, Suberitida, Poecilosclerida, Dictyoceratida, and Cleone. As we were identifying and collecting the sponge, I started creating a field guide based on external morphology. What I have done is to highlight diagnostic characters related to shape, color, oscule, surface that a non-expert, a non-sponge expert could notice and could have a field identification. This guide will be made available to the public once our project is completed. We have added 5 genera to the IRL Porifera: Amorphinopsis, Atlantica, Prosuberites, very common in mangrove habitats. Also, Prosuberites grows on bivalve shells and serpulids and different organisms that have calcium carbonate shells. Scopalina hispida, that is a mangrove species, but it was encountered in an artificial substrate, as well as Spongia tubulifera and Chondrosia affinity reniformis. We have at least 9 potential new species or unknown variants of known species. Very interesting Haliclona species associated to mangrove, a species of Calinula living at extreme low salinities. Very awkward morphologies of Geodia species, the same Yersinia species, a very interesting, widely spread species of Terpios here at the bottom left, associated to artificial substrates or seashells, a sister species or a distinct morphotype of the fire sponge, Tedania ignis. And to solve these studies, we will have to go into the molecular and into deep morphological comparative studies. Why should IRL stakeholders care about this? Well, we must remember that sponges are source of food and habitat for hundreds of species, from mollusks, ophiuroids, lobster, shrimp, polychaete, fish, and turtles. And even manatees, they either eat or they live on or associated to the tissues or on the environment created by the sponges. Sponges are very powerful filter feeders. They filter large amount of waters, and due to billions of bacteria associated to their tissues, with very diverse phyla of microorganisms, make them to play major roles at nitrogen, carbon, silicate, phosphorus cycles. Really, sponges are water transformers. Another important aspect is their role in bioerosion, and particularly in the Indian River Lagoon. At Mosquito Lagoon, there's been extensive research in the past 20, 15 years showing that Cleona celata species affect oyster health, oyster reef structure, therefore the restoration success. There's a very nice poster in the group 4 of the posters in this conference by Harris and Walters that you could study. We are starting to process our bioeroding sponges, and there will be very interesting results in the next presentation. Last but not least, their role: sponges produce large diversity of marine natural products with potential importance for biomedicine. This has not been very deeply explored with IRL sponges. So we have shown you that sponge richness on the IRL is much higher than previously expected. There's a potential 10% of unknown species that might represent local adaptations to unique conditions in the IRL. And we believe that understanding the biodiversity of sponges will contribute to projects on ecosystem health monitoring, habitat restoration, water quality improvement, and fisheries in the system. With that, I want to thank our funders, the Global Genome Initiative, Harbor Branch Oceanographic Institute Foundation, our supporting institutions, especially thanks to Dr. Valerie Paul, and in Harbor Branch, deep thanks to our Jimmy Nelson and Matt Roy for their assistance in the field. With that, I'm finished. If there's any questions.
Thanks, Christina. That was a great presentation. I know a lot of people that are going to be really interested in this field guide when it comes out, and will find it useful.
Great.
We have time for a couple questions if anybody wants to put them in the chat. Chris did a very good job with staying on time, so.
Well, I'm happy. This is incredible.
I'm just going to hang out here for a few seconds to see if anybody's got questions before we move on. Okay, we have one question from Shirley, and she asks, do you anticipate more discoveries during the next field season?
Definitely we do, because we know we visited 25 locations, and although our geographic range was extensive, there's a lot of areas that we were not able to visit. But also the winter, the changes that will be introduced by the season, the water temperature and nutrient conditions, we expect to have definitely more discoveries.
Okay, we have one more quick question. Have you categorized those found on oyster reef specifically? We have a hard time identifying some. This is from Olivia.
Well, we find a very large diversity. For example, we have found several Suberitidae, Terpios, SP, Prosuberites, so associated to these to oysters or other type of bivalves. And it's clear we must be conscious that these Cliona species sometimes represent complex of species. That has happened with the studies of Cliona celata, and you will see results that Iris will show you. So we really need to be carefully compare the morphology, do comparative morphology and genetics to discern really these populations and the role they might have on the oysters, reefs, survival and growth.
All right, thanks, Christina. We're going to move on with our next sponge biodiversity talk from Iris Segura Garcia, and she's going to be telling us a little bit more about the molecular side of things.
So thank you. Well, this talk is intended to be the extension of my colleague Christina just showed you on our efforts to unlock the sponge biodiversity in the IRL, but using genetic tools. So genetic, so genetic markers can assist in DNA taxonomy, as these organisms show or exhibit high levels of plasticity, but at the same time morphological simplicity. So the morphological examination in combination with a genetic barcode can help to distinguish and to identify species. So here I'm showing you two common examples in sponges. So in case one, we have three forms and colors of different specimen sponges that we find in the IRL in this summer collection. So the morphological examination in combination with a DNA barcode help us to conclude that all these specimens correspond to one single species, in this case, Micalia angulosa. Another example is a little bit opposite. We can find specimens of sponges that look very similar in the field, and morphological examinations do not provide enough elements to distinguish species. So these species, in addition and in combination with a genetic marker, can help us to distinguish the presence of protected species. So for our project in the IRL, we want to use at least 2 different genetic markers. We are using 28S, which is a nuclear marker, and it's commonly and very successfully used in the phylum Porifera. And actually, public databases are very well populated with sequences for this gene. We will use this in combination with the standard barcode gene, which is a cytochrome oxidase subunit 1, which doesn't have a good representation in public databases compared to the 28S. So our summer collection is still in process. We collected 261 specimens. I'm still working on those, but I have some interesting results to share with you this morning. Here, I just want, with these pie charts, I want to contrast the performance of the 2 genes. Here in circle, you can see the sequences, the percentage of sequences that match the identified species after a very exhaustive and thorough morphological examination. And we have a very good matches using the 28S information with 61%, compared to only 51% of using the CO1 information. So that is because there is not a good representation of this gene in public databases. And the same low performance was for specimens that were only identified to a genus level, and we have ambiguous calls. So for the rest of my talk, I'm just going to show you the information or the results that we have for CO1, as they are more novel and kind of controversial. So based on the CO1 sequences, we were able to identify some specimens to a species taxonomic level, and with a high confidence. As you can see in the number here, the percentage is the percentage of identity to other gene bank records that are available. And all these identifications were supported by morphological examination and actually the 28S sequences. These species are very common in the IRL, but other species are very common in the IRL, but we don't have reference sequences to identify them. Is 2 species of Micale angulosa, which I showed you an example earlier, and Micale microstigmatosa. The alignment of our CO1 sequences to gene bank records return very low percentage of identity to other generic species, such as Micale cecilia from the Pacific Ocean and Micale marshalli from the Eastern North Atlantic, which means with the information available up to today in public databases, we could only identify our specimens to the genus level. And that is because there is no CO1 reference in the public databases. So that means our records, when we submit them to GenBank, will be the first public record for CO1 for this species. As for ambiguous calls, we have several examples for ambiguous calls, but this morning I just want to share the case for Imeniacidon heliophilia. This species is very abundant and very across all the IRL. We probably see them in most of our sites. Our alignments to public records return very high and equal percentage of identity to other congeneric species, Imeniacidon perlevis and Imeniacidon synapium. So that means that the CO1 does not provide enough information to really distinguish between these 3 species. And as you can see in this box here, which is a fragment of the alignment of the CO1 sequences for this species and others from Florida that I found in the sponge barcoding project here in the bottom. In the top, we have the IRL sequences, and here in red, synapium from the Pacific Ocean and heliophilia from Brazil. So they are basically identical sequences, and with asterisks here in the top indicate that they are the similar nucleotide across all the sequences, and we just have 1 mutation. So we don't really have enough information on variable size, diagnostic size to really distinguish between the species, or maybe these 3 species are the same one. So, and this is in congruence with a recent study that suggests that perlevis and synapium should be distinguished as synonymous species of heliophilia, and heliophilia should be recognized as a globally distributed exotic species. And this is after a very exhaustive morphological and genetic examination that Turner did. So that means that we have a very well distributed invasive species across the IRL. As another example of the lower resolution of the CO1 sequences is the case for excavating sponges in the species in the genus Cleona. We still working on the morphological examination of these specimens. So here in the top line, I'm providing you the field that we come up with the summer collection. So we have some specimens of Cleona celata, some potentially the wind-five Cleona lampa, and potentially Cleona truti. Our alignments to GenBank records return very low percentage of identity to other Cleona celata sequences, even for the sponges that were identified as Cleona celata. There is no identity to Pione, so other species are Pione lampa, and very weird, there is a higher percentage of identity to another conspecific that is distributed in the other side of the Atlantic. So when I reconstruct the phylogenetic relationship of our sequences compared to other CO1 sequences for Cleona celata, the 4 clades that were suggested to be cryptid species from the eastern North Atlantic, here clade 1 to 4. Some of our specimens grew very close to clade 2 of Cleona celata. Very interestingly, our potentially Pione lampa format and different clade, but still within the Cleona celata complex, which is still, but we still need to conduct some other phylogenetic analysis to resolve these phylogenetic relationships. So I include in these 3 other conspecifics just to confirm the distance between Cleona viridis to our Cleone specimens. I included Pione lampa here in the bottom of the tree, as I thought it was going to be very well related or closely related to our potential Pione group, but they are not even closely related. But this relationship and actual identification is likely to be inaccurate, as Pione lampa was described for the northwestern Pacific, actually in Bermuda, and these specimens were from the Red Sea, so it's likely to be inaccurate. So we're still working on our project and our results, but we still many things to still working and analysis to do, but we have some preliminary outcomes. We have new records of CO1 for 2 species of Micali in public databases. We identify invasive species that potentially come from Europe, and we have and we expect to have more species complexes such as the cleonids. And with that, I just want to thank our funding and logistics support. And here is my email if you want to shoot me any questions about methods or dig into our results. And I also want to thank our lab assistants, our master student in the project, that will have a 3-minute talk later. Don't miss it. That's all I have for you guys. Thank you.
Thanks, Eris. We have time for one very quick question if anybody has anything. Not seeing any questions in the chat yet. Okay, let's move on to the next talk. But if you have any questions for Eris, please don't hesitate to put them in the chat, and she can answer them as she is able. All right, the rest of our presentations for this session are actually going to be student presentations. So those of you who are judging, take note. Our first one is going to be from Brianna Cahill, and she's going to talk to us today about a comprehensive diet description for the white-spotted eagle ray. So go ahead and take it away, Brianna, whenever you're ready.
Awesome. Thanks, Hal, for that introduction. And without further ado, I'll jump right into it. So the white-spotted eagle ray is of the family Aetobatidae, meaning pelagic eagle ray. They are listed as near threatened by the IUCN Red List, and they are protected in Florida coastal waters. They do exhibit variable migratory patterns, with northern extents reaching up to North Carolina and southern extents reaching down to Brazil. However, DeGroot et al. 2021 found that the populations in the Indian River Lagoon are fairly residential, whereas populations in the Gulf tend to be more migratory. However, given their pelagic nature, they can encounter fisheries in both Mexico and Venezuela. These rays are of particular interest because they have this hard plate-like dentition that allows them to effectively consume bivalves, gastropods, and potentially crustaceans. And they're also very effective at excavating their prey by using their pectoral fins in order to liquefy the sediment around them and burying their head in order to access burrowing bivalves. So we're wondering why we're interested in understanding the diet. And it's because we don't know a huge amount about these rays. While their diet has been described in Mexico and Bermuda, we really don't understand their diet and their trophic ecology in Florida coastal waters. And especially given how much shellfish enhancement occurs in Florida coastal waters, whether it's shellfish aquaculture or shellfish restoration, this is gaining insight into their potential interactions with these shellfish enhancement areas. So we have three main objectives for this study, first of which is to characterize prey importance to the diet of these rays via their stomach contents. The second of which is to assess stable isotope tissue variation for these rays. And then finally, to assess variation in stable isotope values of their prospective prey items. So there are two main sampling methods that we will be employing for this study. The first of which is DNA barcoding, which will be used to supplement visual identification of their immediate diet or their stomach contents. And this is because sometimes the pieces are too small, hard to identify, or they're digested. And so using DNA barcoding as a gene-specific method, we will be using the CO1 region, as Iris just talked about, in order to potentially identify those different tissues. And the premise is that we're able to take those unidentifiable tissues, extract the DNA, sequence it, and then hopefully compare it to information available in barcode databases like GenBank and Barcode of Life. That way we will be able to identify those stomach contents. The second method we will be employing is stable isotope analysis, which is a chemical and ecological approach in order to understand their longer duration assimilated dietary habits. And so using nitrogen and carbon, we'll be able to track their trophic level and basal carbon source. And this is extremely beneficial in the absence of stomach contents, as not all of the rays that we've sampled so far have had stomach contents. So we're still able to collect some information from them. So far we have been targeting rays in the Indian River Lagoon and over in Sarasota, Florida. And so we do that by going onto the boat and visually spotting rays. And then once we spot them, we set a large net around them and try to corral them. That way we can get them into a live well on the boat. Once they're on the boat, we go through a slew of morphological measurements, and we collect blood and muscle biopsies for stable isotope analysis, and then finish it off with pulse gastric lavage, which is a non-lethal means to collect stomach contents, of which we're able to assess at a later date. Another important component of this project involves prey collection, which for the most part, we've been targeting via accessible sandbars and shallow benthic habitat. However, we have also opportunistically collected while diving. And there are three main goals for these prey items. The first of which is to develop a thorough visual guide as to whatever prey items we can find. That way, when we're processing the stomach contents, we have a higher likelihood of being able to visually identify these prey items. The second of which is to supplement CO1 information that's already available in GenBank and Barcode of Life. However, some of the species that we've collected so far have not had their CO1 region sequenced. So we're hoping to take this information and be able to use it. That way we have a higher likelihood of positive identification when we're sequencing the unidentifiable stomach contents. And then finally, we will be using that remaining tissue for stable isotope analysis. That way we can generate mixing models in order to retroactively determine eagle ray diet. As far as stomach contents go, from 2020, we have collected 26 samples, and then so far from 2021, we have collected 4. However, we have not been able to fully process all of them just yet. So I will be talking about these 15 eagle ray archive samples that we have from 2015 to 2018. So as you can see, just to orient you to this plot, we have our 15 different eagle rays considered at the bottom, and then we have the proportion of positive identification of stomach contents, not necessarily proportion of diet. But what you can see is that there is quite a bit of variability in between these 15 different rays. However, there are 3 species that are found in at least a third of the stomach contents, of which include the sunray Venus clam, the banded tulip, and the Florida fighting conch. Additionally, we wanted to look at how these positive identifications can differ in regards to eagle ray disc width size. And so there are 3 species that are found across a wide size range, including the sunray Venus, again, the banded tulip, and then the lettered olive, of which tend to either have thinner shells or are, for the banded tulip and the lettered olive, tend to be smaller gastropods. So a little bit easier for smaller eagle rays to be able to fit them in their mouth. And then conversely, when we're considering some of the thicker shelled bivalves, thicker shelled gastropods or larger gastropods, we're only seeing them in the larger eagle rays, of which include the Atlantic surf clam, the Mercenaria hard clam. We're also seeing lightning whelks, which tend to be a little bit of a larger gastropod, And then the Florida fighting cock, which tends to get thicker as it gets older, and can tell you from experience they're a lot harder to crack into than a lot of other gastropods. Regarding stable isotopes, currently we have samples from 43 different individuals ranging between having muscle, plasma, and red blood cell samples. Additionally, we were able to take advantage of some archive data that we had from the Atlantic coast between 2017 to 2019. So that's in part a big reason why there's such a discrepancy between the number of samples from the Atlantic coast and the Gulf Coast. Moving forward, we'll be processing more 2020 samples and 2021 samples as well. Regarding the tissue types, we are starting to see some slight differences in between the 3 tissue types considered. For muscle, we're seeing that they have higher nitrogen and a higher carbon value, which could be a result of the muscle having a longer isotopic turnover rate, therefore incorporating more assimilated dietary tissue. And then for the plasma, because it has a much faster isotopic turnover rate, that's my assumption as to why it might have a lower nitrogen value. But as we continue to incorporate more specimen, we'll be able to run statistics and be able to tell you on a more solidified note. Additionally, we wanted to look at the 3 different tissue types in between the 2 coastlines considered. And again, we have a smaller sample size for the Gulf. However, it does appear as though the nitrogen is slightly low from the Atlantic to the Gulf side, and carbon is slightly higher on the Gulf side, which could reflect a difference in prey consumption, but also foraging in different ecosystems. And then finally, we have 14 different prospective prey items that we've sent off so far for analysis. However, they all have a very small sample size, so we can't run any statistics just yet, but we are starting to see some general patterns emerge in which bivalves have lower nitrogen and lower carbon values, likely because they're feeding on phytoplankton. Our crustaceans have a middle range nitrogen value, likely because they are detritivores, and so they're foraging on whatever they opportunistically can. And then we're seeing our carnivorous gastropods at the highest level with the highest nitrogen, likely because they're foraging on bivalves and have the predatory role in this plot. And then we also have our one herbivorous gastropod with the highest carbon value. So some preliminary conclusions that we've observed so far. There are some species that are observed across all size ranges for these stomach contents, which include the sunray Venus banded tulip and lettered olive, whereas the larger eagle rays tend to be the ones consuming these thicker shelled bivalves or thicker shelled gastropods. We also saw a very limited consumption of crustaceans. We only had 1 positive identification, but I can tell you from our 2020 samples, there will definitely be more of that moving forward. And then finally, we are starting to see some discrepancy in between the tissue values for these stable isotopes. And so this is very important for us to understand is, as I described, we did find Mercenaria hard clams in the stomach contents. And like Todd Osborne hit on yesterday, we know that the natural stock of hard clams is fairly hard to come by. And so it's possible that these rays are interacting with shellfish enhancement. And so if they are, in fact, we need to develop strategies to prevent predation. Finally, additionally, because these rays are reliant on benthic communities, we need to understand their energy flow and how they could be negatively affected by coastal development and harmful algal blooms. And then building off of that, we need to understand how their diet could be a possible source for toxins, leading to biomagnification within the food web. And so with that, I just love to say thank you to my lab, my external support, and all of my funding sources.
Great. Thanks so much, Brianna. That was great. We have time for one really quick question. I'm just going to go with the first one. Michelle is asking, where are spotted eagle rays typically found in the IRL?
We can actually find them. For the most part, we target the inlets just because when it's an incoming high tide, the water's a lot clearer, so it's a lot easier to see them. But my advisor has seen them pretty far up into the lagoon, just around some of the spoil islands. They're definitely around. It's just a matter of can we catch them in a convenient spot.
All right, well, thanks, Brianna. If you have any more questions for Brianna, feel free to put them down in the chat and she can answer them as she's able. We're going to move on to our next student presentation from Jeffrey Herr, and he's going to be talking about diamondback terrapins in the central IRL. Take it away, Jeffrey.
Hi, So this is an update on my diamondback terrapin research. So before we get any further, let's see, why is my computer, there we go. So just what are diamondback terrapins? They are a small brackish water turtle. They live along the east coast of the United States from Cape Cod to Corpus Christi. There are 7 recognized subspecies, and we have 5 of these in Florida. The subspecies I'm going to be focusing on today is Malaclemys terrapin tequesta, or the Florida East Coast diamondback terrapin. Prior to the 19th century, diamondback terrapins were extremely common, to the point where they were used as pig feed. And people started eating them. By the late 1800s, it was an extremely popular food item, and there was a very large fishery. This, of course, caused a collapse of the terrapin population. And around Prohibition and the Depression era, the terrapin food fad ended, and diamondback terrapin populations began to recover. Diamondback terrapins are listed as vulnerable by the IUCN Red List. However, they lack federal protections in the United States. They're still in decline across their entire range, and they suffer from habitat loss, crab trap, bycatch for blue crab fishery, and collection and poaching for the pet trade. Within the Indian River Lagoon, diamondback terrapins were extensively studied in the Northern IRL in Merritt Island National Wildlife Refuge in the late 1970s. This population was revisited in the 90s, and what once was a population estimated around 600 was estimated around 9 individuals. There was a massive population collapse in this time. Since then, a little work was done here and there, but really after 2014, and a paper was published challenging the subspecies designations and designating all Florida terrapins as a single management unit. So, but this paper didn't include any samples from the east coast of Florida, including, of course, the Indian River Lagoon area. So this renewed interest in diamondback terrapins in this region. So FWC conducted a multi-year study in the Banana River that I believe they recently concluded. They collected 200-something terrapins and took tissue samples and did market recapture studies with them. And then I've been doing some work along with some collaborators in the Central Indian River Lagoon. So in the Central Indian River Lagoon in 2016, some diamondback terrapins were found on land trust property around Vero Beach. So this led to a preliminary study involving a citizen science project, bringing a summer camp out there. They found 14 individuals, they took tissue samples, and they marked these animals with shell notching for recapture. I took over this study in 2018 and started trapping in 2019. I found 15 new individuals that year. I didn't have any recaptures from 2016. I implanted pit tags rather than using shell notching, took tissue, blood, and fecal samples, and acoustic tagged 12 terrapins for passive tracking using my receiver array and fact network. This year, or the previous year in 2020, I found 12 new individuals, and I also recaptured 3 individuals from 2019. Again, these animals had the same procedures done to them, and I deployed 10 acoustic tags in 2020. So part of the project is acoustic tagging and movement. I have 7 receivers deployed around the study site, including 1 receiver in an internal pond. I was able to tag 12 terrapins in 2019, and I deployed 10 tags in 2020. 2 of these tags were on terrapins who had previously been tagged, but the battery on their tag had died by the time I recaptured them. Another terrapin was tagged early in the season. Based on injuries she sustained, we believe she suffered a boat strike and lost her original tag, and she was retagged towards the end of the season. So part of the goal for this is to determine if the population is localized or if the population is free-ranging. These terrapins have exhibited very high site fidelity. Most of the terrapins tagged in 2019 were present at the study site months after being tagged, and 3 terrapins from 2019 were recaptured again in 2020. I also had a lot of recaptures over the several months of the trapping season, including the terrapin pictured on the previous slide, who I caught at least 8 times. Next, I was doing a hematology project with the help of one of my students at the Harriet L. Wilkes Honors College. We calculated packed cell volume and sent off the samples for a blood chemistry panel and protein electrophoresis. I don't have time or space to show you all of the results from here, and we did not have a very high N, so all this is preliminary, but we found some correlations between salinity and certain blood chemistry parameters, including packed cell volume, total protein, albumin, and alpha-2 globulin. Next, we were doing a dietary study. So we took 13 fecal samples from 10 terrapins, and we sorted through these samples and grouped what we found into 7 categories. Decapods, bivalves, vegetation, barnacles, gastropods, fish, and unidentified. So here in the upper corner, you can see a fecal sample very shortly after coming out of a terrapin. And in the middle, you'll see a protoconch of a gastropod. The next slide image is part of the carapace of a crab. And the bottom one is a unidentified gelatinous substance that we found in large quantities in one of our samples. So what does that tell us? Well, we compared these to other diamondback terrapin dietary studies. And mostly we were comparing it to this 2016 study from the Southwest Florida and Everglades area. They found that plants were the most commonly eaten food item and gastropods were the second most common. However, in our study, gastropods were extremely uncommon. They were actually only found in 1 sample. So I'm not really sure what to make of this. We're collecting more samples this year, and hopefully we have some more solid results to report. I'd like to thank all of my collaborators, my students at the Honors College, and everybody in IRLAP who's helped me, the funding from the HBOI Foundation, and the FACT Network and Indian River Lagoon Land Trust. So thank you, and I hope I have time for questions.
Thanks, Jeffrey. You do have time for questions, and we have one for you. Susan wants to know, did you find any microplastics in the terrapin's fecal samples?
So that was one thing we were looking for. My original hope is that I'd be able to process the samples similarly to the way oysters have been processed for microplastics. But that generally is a destructive process, and I need to be able to see the contents. Once we filter out the contents so that we can see the samples, then I'm unable to do the microplastics analysis. So at some point, I'd really like to do that, but I'm not sure if that's going to be possible.
All right, thanks, Jeffrey. If you got any more questions for Jeffrey, please feel free to put them in the chat. We're going to move on to a talk from Marissa Pico, and she's going to be talking about a non-invasive technique for estimating body condition in green and loggerhead sea turtles. So whenever you're ready, Marissa.
Thank you so much. I will be talking about my thesis, which I'm a master's student and it's titled Validating Ultrasonography as Non-Invasive Technique for Estimating Body Condition in Green and Loggerhead Sea Turtles. And I am a part of the Page Cargen Lab. Some of the sea turtle threats that we find in our Florida waterways are fibrobapillomatosis, which you can see in the top left picture. It's a neoplastic disease or virus that promotes these tumor growths, as you can see, as the pink tumors that you see on the turtle in the top left. Also, there is boat strikes. Boat strikes are commonly found around Florida waterways and are usually typically very fatal, as you can see in the bottom left picture. Sometimes the strikes will actually open the coelomic cavity. The top right picture shows microplastics and plastics as a form of a threat. Sometimes sea turtles will think that plastic bags are jellyfish and eat them, and then they will end up choking on it. And then another threat is the harmful algal bloom. It doesn't directly kill them, but it does kill them. It kind of kills the seagrass off, and seagrass is the main diet for the green sea turtle. So that's typically the type of threats that we find around Florida coasts that are affecting our sea turtles and their overall body condition. So because their overall body condition is being threatened, we have started conducting health assessments of the green and loggerhead sea turtles around Florida's coast. So sea turtle health assessments are an integral part of determining a population's overall health and recovery efforts. So a few procedures happen during these health assessments. Once we capture the sea turtle, we do body morphometrics, which is basically measuring the carapace and weighing them as well. There's a picture of that in the bottom left-hand corner. We take some clinical blood data. You can see that in the second to left picture. And that typically encompasses PCV, which stands for packed cell volume, and total solids. We also do a skin and scoot sample, which you can see in the second to right picture. And that is usually for isotope analysis, that is for separate studies. Finally, we do a body condition evaluation, and that encompasses a visual subjective body condition score, calculated body condition index, and my study, which is the ultrasonography. And you can see that in the bottom right picture. So I'll go a little bit more into body condition score, which is a part of the body condition evaluation. Subjective body condition scoring basically is a ranking from 1 to 4, 1 being emaciated, 2 being thin, 3 being good, and 4 being robust. So we kind of just look at the turtle and we can give them a score 1 through 4. So a turtle that would be emaciated would be the turtle in the bottom left-hand picture. And you can see that the eyes are like sunken in. There's really no fat around the neck and shoulders. And if you would see them upside down, their plastron would be concave inwards. And then the picture second, right after that, would be a turtle that is thin. So he would receive a body condition score of 2. He is not as skinny as the first one, but he still is lacking some of that fat abundance. A turtle with a body condition of 3, which is good, would be right after that one, second to the right. And they're basically a very healthy turtle. And then finally, a robust turtle, it's basically another way of saying a fat turtle. They're also healthy, and a picture of that would be in the bottom right-hand picture. The problem with body condition scoring is that because it's subjective, there seems to be a lot of variability between evaluators. Next, we have body condition index. So this is a more precise way of doing it because you need to use a calculation. So for this calculation, you need to have the weight, which you can see them doing in the bottom left-hand picture. It can be really cumbersome, especially for turtles weighing hundreds of thousands of pounds. And you also need a straight carapace length, which you see them doing in the bottom right-hand picture. So this one, most people will get very similar answers, unless you do your measurements and weights a little off, but it's more precise in that way. However, for those leatherback sea turtles that weigh thousands of pounds, it can be very cumbersome. So that's where my research comes in, ultrasonography. We want to use and compare 2 different portable ultrasound machines, which you can see in the top. We have the IBEX Evo and the Renko Lean Meter. And they're to measure subcutaneous fat in the green and loggerhead turtle carcasses. So that's the first objective of my study. And the second one was for live captured sea turtles that we found in Florida waterways, was to evaluate the relationships between ultrasound assisted shoulder fat depths, calculated BCI, subjective BCS, packed cell volume, and total solids. However, the fourth objective of my study, which I will be going more in depth in this talk, is to develop those reference quartiles for BCI based on the relationship between calculated BCI and subjective BCS data assigned for live captured and stranded turtles. This is really important because once you have like a BCS or a BCI, you should easily be able to tell vice versa what their body condition is overall. So for that part of my study, we used green and loggerhead sea turtles of all age classes. And we basically just collected data from rehabilitation centers and research groups all across Florida. So we used Loggerhead Marine Life Center. We also used Georgia Sea Turtle Center, a few studies on debilitated loggerheads, and a study on Florida live captured sea turtles. We got a lot of sea turtles from them, but the majority of our sea turtle data collection came from In-Water Research Group, and they're located at the St. Lucie Power Plant. And they gave us about 15,000 data points that dated back to 1987. So with all of that combined, we had about 15,700 total data points that we can use for the reference quartiles. So to analyze that, for the loggerhead and green turtles, we assess the BCI values alongside the BCS to see if there's any sort of relationship. And then we put them in different quartiles based on the 4 BCS ratings, which I mentioned before: emaciated, thin, good, and robust. So we separated them based on species, and upon initial plotting of a graph, you can see that there are 4 different conditions. They are separated, but however, there is this area right here that shows there's BCI scores that fit into all 4 categories. So it's important to know, but that does cause a little bit of concern that sometimes they're not adding up correctly, but that just could be error either in BCS scoring or body condition calculating. However, based on the analysis I did, I put them for loggerhead turtles into 4 quartiles, and for turtles with BCS of 1, which are considered emaciated, they would have a BCI of less than 1.17. Turtles with 2, which meaning they're a thin condition, should have between 1.18 to 1.32. A BCS of 3, meaning good, should have a score of 1.33 to 1.41. And then a fat turtle, which would be robust turtle with a BCS of 4, would have greater than 1.42. And then as you can see, the green turtle results were a bit different, so that's why we separated them to begin with. But you can also still see that there are 4 separation groups, but there is that still small area where some of the BCI numbers did fit into all 4 categories. So that is something we definitely need to look into more, but it's good to know. As for the quartile rankings for green turtles, turtles with a BCS of 1, meaning emaciated, should have a BCI of less than 1.04. For those turtles with a BCS of 2, meaning thin, should have a BCI of 1.05 to 1.19. Turtles with a BCS of 3, meaning they're a good condition, should be 1.20 to 1.29. And a BCS of 4, meaning robust, should have a BCI of greater than 1.30. So some future projections with this study, it would be really good that we can analyze the health of the turtles at each of the quartiles and relate it to their clinical blood data and epibiota load. So for example, looking at those turtles, PCV stands for packed cell volume, and their total solids, seeing how that relates to their body condition, as well as barnacle load, leach's load, and any fibroblastic papilloma tumors, see how that relates to their overall body condition score as well. Also, this is really important because veterinarians and sea turtle biologists will have a way to quickly estimate body condition. Say they're on field day and on the boat or on the beach, and they need to do something quickly. And this could be really helpful for them in the future. And here are my references, and I will take any questions. These are some images from conducting my ultrasonography research.
Thanks, Marissa. We are running short on time, so we need to keep moving, but I know you have, looks like at least 2 questions in the chat, so feel free to get to those when you have a chance.
Okay.
Thank you. And our next and final presenter for this session is going to be Jessica Copertino, and she's going to be talking about a survey of bird abundance and diversity in the Mosquito Lagoon.
Thank you. So can everybody see my screen?
Yeah, we can see it. You're just not in presentation mode yet.
It's not, there we go.
There we go.
Okay. So I'm really excited to share with you all today some of my thesis research on bird abundance and diversity in Mosquito Lagoon. So first, just a little bit of background. In dynamic ecosystems such as the IRL, it's really important that we monitor biodiversity, and this will help us to detect population declines, particularly for threatened and endangered species, to assess impact of environmental changes, and also evaluate and assess the successes of restoration efforts. And in previous studies, birds have been used as indicators of ecosystem health and have also been used as proxies for biodiversity. However, in the IRL, most of the bird research has been occurring in the Kennedy Space Center and Merritt Island National Wildlife Reserve area, with very little focus in the more northern portions of the IRL, like Mosquito Lagoon. So my research study objective was to provide data on the abundance and diversity of the avian community within Mosquito Lagoon, with a more long-term goal of being able to use that data to better assess the impacts of future environmental changes, both natural and anthropogenic. So to do this, we created a 14-kilometer transect route, as shown in the map to the right in bold. And this 303-hectare route includes a myriad of different habitat features, including live oyster reefs, which includes both natural and restored reefs, shell rakes, which are composed of piles of dead disarticulated oyster shells, mangrove fringes, dead standing trees, artificial structures such as signs, pilings, as well as aquaculture equipment, open water, and also sandbars. So in this 2-year study, we conducted transects by boat at least once per month, and the north-south or south-north direction of the transect was decided haphazardly each time. And we conducted transects at various times of the day and also during varying tidal cycles. Photos were taken of all birds seen along the route within 50 meters of the boat. And then from those photos, we identified every bird to species, and we sorted the birds based on which habitat features they were utilizing. So looking at some of our looking at some of our abundance data first, we observed 21,649 total bird observations, and that was for a total of 67 unique species that we identified. And on average, we observed 416 birds per transect route, which comes out to a density of about 1.4 birds per hectare. Looking at some of the seasonal trends in the graphs on the right-hand side, all of the x-axes on these graphs are date. We're going to look first at graphs A and B, which are abundance and species richness respectively. And if you look at both of these graphs, the abundance and species richness is highest in the winter months and lowest in the summer months. And this is because during those winter months, we have a combination of permanent resident species as well as migratory species that are arriving in the lagoon towards the end of fall and then leaving at the beginning of spring. So we have the highest abundance and species richness in the wintertime. However, if we look at graphs C and D are evenness and diversity, Simpson's diversity. And evenness is basically just refers to how close in numbers each of the different species are within that ecosystem. So we see here that the evenness is actually lowest in winter and highest in the summer. And this is due to the predominance of those migratory birds during the winter seasons. For example, in one particular winter transect, 65% of the birds that we saw were red-breasted mergansers. So the evenness values during those winter months are really low because of those migratory species predominating in that particular time, whereas the summertime we have a higher evenness. So therefore, our Simpson's diversity is also higher in the summer months because Simpson's diversity is a bit more sensitive to changes in evenness than to changes in richness. Next, I wanted to look at the differences in bird assemblages using different habitat features within the lagoon. So to do that, I created an ordination plot using non-metric multidimensional scaling. So just to orient you, the different symbols on this graph represent bird assemblages utilizing specific habitat features during a particular season. And the closer together two symbols are, the more similar those assemblages are, the farther apart, the more different they are. The ellipses represent 95% confidence intervals. So wherever we see overlaps in the ellipses, like what you can see between, for example, oyster reefs and mangroves, that represents that there are similarities in those bird assemblages. Whereas if you're looking at ellipses that don't overlap at all, like standing dead trees and floating in open water, that indicates that there are differences in those bird assemblages. I used a Permanova analysis to determine that there are significant differences based on habitat features, signified by our p-value of 0.001. And the stress on this graph, on this ordination plot is 0.17, and typically stress values between 0.1 and 0.2 are said to be a fair representation of that data. So then to dig into this a little bit further, I wanted to see if there were any species that were representative of specific habitat types. So I ran an index species package in R, and it revealed that there were indeed 36 species that were representative of either a single habitat feature or a combination of habitat features. And for the sake of brevity and for the sake of this 10-minute talk, I'm going to focus on just a handful of those, mainly looking at birds that are threatened. So the first indicator species that we're going to talk about is least terns. And these came out as an indicator species for the shell rakes. And with a loss of sort of more typical nesting habitat for least terns, which would be beach nesting habitat, they're nesting even more predominantly on shell substrate in their nesting range in Florida. So we did see nesting least terns on 2 separate shell rakes along the transect route throughout the study, throughout the summer months, spring and summer months of the study. Next, we have indicator species for live oyster reefs, which was a combination of both natural and restored reefs. So American oyster catchers came out as an indicator for this particular habitat feature, and this is likely due to the fact that they are foraging directly on the live oysters on these reef types. 2 of the other species that came out as indicators were black skimmers and reddish egrets. Their primary food sources of these 2 species are small fish, and the reef complexity created by these oyster reefs is providing habitat for those small fishes. 2 indicator species for sandbars were the semi-palmated plover and the roseate spoonbill. Semi-palmated plover's main diet is composed of things like small gastropods and polychaete worms, which can be found in abundance both on the surface of the sandbar as well as in that sediment. Whereas the roseate spoonbills, they were mainly found along the edges of the sandbars, where they could put their bills into the water and sweep them from side to side throughout the water and the soft sediment on the bottom to search for prey such as fish and other marine invertebrates. The indicator species that came out for the combination of mangroves and live oyster reefs were mainly wading birds with several species of herons and egrets included. So here we have pictured a tricolored heron in both photos here. And also little blue herons were indicator species for this combination of habitat features as well. And these herons typically feed on small fish, as well as shrimp and decapod invertebrates, and the prop roots of the mangroves, as well as the shell complexity of the oyster reefs, provides perfect habitat for those prey items, which may explain why so many wading birds were in this category of indicator species for these 2 habitat features. And then lastly, we have the osprey, which was an indicator species for the combination of artificial structures and standing dead trees. So both of these structures would provide aerial perches for these birds to both search for prey and to return with their prey to consume it. And it's interesting that these pilings and signs may even be providing a better place for the birds to look for prey because it's actually directly above the water. And then they also don't have to travel as far with their prey and potentially avoid dropping it by going to these pilings rather than flying all the way up to a tree. So now some of the conclusions and future directions here. So it appears that the mosaic of habitat features in Mosquito Lagoon does support a diverse bird community. And due to the high trophic levels of birds and their ability to seek out and use suitable habitat, birds are good indicators for restoration success. And we know that there's a lot of restoration efforts ongoing in Mosquito Lagoon. So this data will be important for assessing ongoing and future restoration projects within Mosquito Lagoon. And with that, I'd like to acknowledge my funding sources, as well as all of those individuals who have been helpful in this study. And I will take any questions.
Thanks, Jessica. We have time for one quick question before we need to move on, if anybody has anything for Jessica. Not seeing anything pop up in the chat right now. So we're just going to move on to the 3 minutes and 3 slides section. Oh, there is one coming up. So you'll have some that you'll have to answer. Thank you so much to all of our presenters in this session. Thank you to those of you who have had questions. And we will move on to 3 minutes, 3 slides that is being chaired by Zach Judd from Florida Oceanographic Society. Thank you.
All right. Well, thanks, Holly. And I'm excited to introduce our final round of 3 minute, 3 slide speakers. I'm also sad that this is our final session for the 2021 IRL Symposium. I want to give a big thank you to all of our presenters and poster presenters this year, as well as our event organizers. I hope all of you will agree, the symposium was a tremendous success despite the transition to virtual format. I want to remind everybody we will be using the chat box for questions during the 3 minute presentations. And we're going to get started with Greer, who's going to tell us about IRL sponge communities.
Thank you. So to get into it, do sponge communities reflect a biome transition zone in the Indian River Lagoon, or IRL? The IRL's geographic location is clearly a transition zone between subtropical and temperate climates. Although sponges are valuable organisms, little is known about the diversity of sponges here in the IRL. Last summer, a team of scientists braved the COVID and collected sponge samples at various places throughout the IRL. I took a subset of the resulting presence-absence data and ran multivariate statistics to test the differences in sponge communities at 19 sites to see if we do see sponge communities reflecting that biome transition zone. The map on the left has 19 points. Each point is colored based on the habitat, and you'll notice that 19 sites points are organized in 4 general categories: Mosquito Lagoon, Banana River, Central IRL, and Southern IRL, moving north to south. Okay, next slide. Here is a cluster dendrogram looking at all of our different locations and sites. You'll see at the very far left, there's a yellow bar, and this is our Banana River site. I ran a 2-way Permanova looking at locations, differences in sponge communities at locations and habitats, and found significant differences in our locations. Looking at the bottom right, you will see our SIMPR results, and you'll notice that the Banana River location was driving that differentiation, and specifically 2 species of sponges. We have a Calanula species and a Prosuperiotes species. You'll also notice at the far right of our cluster dendrogram a cluster of our muddier habitats. Although there was no significant differences amongst habitats, we do see this clustering. Next slide. Here at the top, you will see 2 NMDS plots. NMDS, very quickly, the closer the points are, the more similar they are, and the further away, the more different. You will notice at our left top NMDS plot, the Banana River location is completely isolated from all other locations once again. At the bottom left, you will notice that the salinity at this site was very low compared to our other sites, and we think that's why we might be seeing different species at that particular location. At the top right, you will notice an NMDS plot eliminating the Banana River location, and you'll see that the remaining locations are overlapping. I did run a second two-way Permanova and found no significant differences between locations. So in conclusion, differences between locations were driven by the Indian River location. However, we do hope that when we add more data to these statistical tests, that we might see the sponge communities reflect our biome transition. Thank you.
Fantastic. Thanks, Greer. Our next speaker is Megan, and she's going to tell us about the Harbor Branch FAU Sponge Cell Culture Collection.
Thank you. All right, so sponges are diverse components of marine ecosystems and are a prolific source of chemicals with potential human health applications. We have established methods to cryopreserve sponge cells, developed the first marine invertebrate sponge cell lines, and created the first frozen biobank of living marine sponge cells. Over the years, we have collected more than 200 sponge individuals representing more than 50 species, 26 families, 15 orders, and 2 classes. The HBOI FAU Sponge Cell Culture Collection can be used as a repository for sponge biodiversity as well as to support research in habitat restoration and biotechnology applications. Next. After an extensive survey and representative collections of IRL sponges, we cryopreserved cells from 65 individuals representing 8 orders of the class Demospongiae. We tested samples in an optimized nutrient medium, and these graphs display the % change in cell number on the x-axis and the different samples tested on the y-axis. The red bars represent artificial seawater, our control. The blue bar is our basal medium, M199, and the green bar is our optimized nutrient media, M1. And we found exceptionally rapid cell division occurred in approximately 20% of the sponges. In the top left graph, we have a large increase in cell number from Niphates erecta and cells from 2 Chectactinellid species as well as Subaritidae species, Amorphinopsis atlantica and Hymeneacidon heliophylla, also divided in our optimized media. We will continue to expand our biobank with IRL samples during the next planned collection in March, and research continues with optimization of this nutrient medium for the IRL species. Next. And finally, the sponge cell culture collection will be used to develop habitat restoration by graduate student Elizabeth Urban Gadamke and biotechnology applications by graduate student Cassidy Dugan. Research is underway to develop a method of culturing sponge cells in a 3D matrix using optimized nutrient media to form differentiated sponge aggregates. We have successfully cultured sponge cells using fiber cell discs, thin hydrogel layers, gel microdroplets, and spheroids. And we found that improved longevity of cultures are observed with 3D methods versus 2D cultures. We are also working to establish a hybridoma sponge cell line through fusion of sponge cells that do not produce any bioactive compounds, but do divide rapidly in the media, with sponge cells that do produce the compound but do not divide in the medium. We're testing the hypothesis that a rapidly dividing hybridoma cell line will be achieved by culturing hybridoma cells in the optimized nutrient medium, and the bioactive compounds will be produced by the cultured hybridoma cells. And that's all for me. Thank you for listening, and don't hesitate to reach out.
Thanks, Megan. Christy's up next, and she's going to tell us about marine natural products from the IRL.
Thank you so much. Can you hear me okay? Awesome. Okay, so we all know that the Indian River Lagoon is one of the most biodiverse estuaries in North America. This biodiversity translates to incredible chemical diversity, which is what I'm interested in studying. More specifically, I researched the specialized metabolites or marine natural products produced in these organisms, which can be used in drug discovery. The goal of my dissertation research is really to identify these marine natural products which reduce levels of survivin in cancer cells and therefore represent potential anti-cancer drug leads. Next slide, please. Survivin is a protein that is upregulated in cancer cells and is correlated with poor patient prognosis. The Wright and Guzman labs here at Harbor Branch developed an assay called an immunofluorescent high-content imaging assay used to measure the levels of survivin protein in cancer cells in response to treatment with marine natural products. You can see here that the survivin protein is labeled in red, and high levels are expressed in normal cancer cells in figure A. After treatment with an active compound, the survivin levels are reduced, which is what we want to see. From this assay, an extract from a tunicate found here in the IRL Eutostoma olivaceum, pictured below, was identified as a potent survivin inhibitor. I purified the compound from the complex mix of chemicals in the extract, as seen here in the HPLC trace in figure C, and determined the structure of this pure active compound. Next slide, please. The next step was to identify how this pure compound was acting in the cancer cells to reduce survivin levels. If we remember the central dogma of biology, it states that DNA makes RNA and RNA makes protein. So if we are observing a decreased level of survivin protein, we may classify that mechanism into two broad categories. Option one, the survivin simply isn't made, meaning the DNA isn't transcribed to RNA, and we can measure that with RT-qPCR. Or option two, the survivin protein is made, but is targeted for rapid degradation, resulting similarly in lower survivin levels. The results of my RT-qPCR experiments in the bar graph show that my pure E-olivaceum compound, labeled 41102, did not reduce survivin transcription, so we can conclude its mechanism is not option one. But when I look at the degradation rates of survivin protein, there is a statistically significant difference at the 12-hour time point of about 50% for treated cells compared to control. This suggests that the pure compound from E. olivaceum is acting downstream to target survivin protein for increased degradation. There's still a lot of work to be done to determine exactly how this compound acts in cancer cells, but I believe this data is a step in the right direction. Next slide. Potentially. And with that, I'd like to thank my committee and lab members for their help and support, and of course, my funding sources. Thank you.
Thanks, Christy. Next up is Lauren to tell us about fine-scale behavior of bull sharks in the IRL.
Okay, thank you. So today I'd just like to introduce a project that we've just started in the Fisheries Ecology and Conservation Lab at Harbour Branch. And I'd just like to start by acknowledging the funders, which are the Save Our Seas Foundation and Harbour Branch Foundation. Next slide, please. Oh, skipped a slide. Interesting. Okay, well, I'll just carry on, but there's a whole slide in between these two. So basically the aims of this study are to develop a tag package, which allow us to look at the fine scale behavior and activity of bull sharks in the Indian River Lagoon. So we know that it's important nursery for them, and we know that they occupy a high trophic level in the Indian River Lagoon. So we've essentially tried to pull together a multi-sensor package that we can attach to the animal that comprises an accelerometer, which measures the activity, the body movement of the animal in 3 axes. A magnetometer, which gives us an estimate of the animal's heading. There's also a passive acoustic transmitter in it, that is giving us an indication of the animal's space use. So I would be able to show this is all on the previous slide, but then there's also a satellite tag as well, which gives us an indication of where the tag has popped off of the animal after about 3 days. So we attach this to the first dorsal fin of the animal, and we need to get the tag back in order for us to be able to download the data. So what's really cool about this tag is that it's giving us a huge amount of data in a really short space of time. So the accelerometer itself is logging 50 data points per second in 3 axes. So it's giving us a really good idea of what these animals are doing. And when we put the accelerometer and the magnetometer data together, we can actually get an estimate of where the animal's moving and do a reconstructed path known as a pseudo track. And that's what you can see on the left-hand side. We can also get an indication of the animal's tail beat frequency from the accelerometer, which is indicated by the colors in the left plot and also on the depth trace in the top right plot. And the other thing that's really cool is the squiggly lines that come out of the accelerometer can have quite unique signatures. And we can use those signatures to actually classify different behaviors and then make predictions on upcoming data sets. Next slide, please. Okay, that was the first slide. So this is what the tag looks like. And we'll just go through to the next slide and see if, there we go. So that's the takeaways. So we're trying to understand the fine scale behavior and movement of the animal in time and space. We want to see if they're responding to harmful algal blooms. And the big thing that I really just wanted to ask people, because obviously there's a lot of people at the symposium that spend a lot of time on the water in the IRL. If you see this bright orange tag bobbing around in the water, please don't just run it over with your boat, grab it for us. As I said, we have to get the tag back in order to be able to download any of the data. There's a phone number on the back, so if you find it, please let us know. We'll be super excited and very appreciative. And we're hoping to put about 20 tags out over the next year, so follow our progress on any of the social media streams listed below. And thanks for listening.
Thank you, Lauren. Sorry about the slide issues.
It's all right.
Our next 5 presentations are all components of the same larger project, a project that looks at potential impacts of restoring lagoon inflow. Our first speaker is Gary. He's going to talk about modeling anthropogenic inflow scenarios.
Okay, I just want to do a sound check. Can everyone hear me? Okay, great. I've been working on these issues for many years, up and down the eastern seaboard of the U.S., beginning way back in 1985 in Great South Bay, which is a sister lagoon, very similar to IRL, except in a cold environment, has some of the same issues. And the preface here is that historically and over geologic timescales, there is natural exchange between these coastal lagoons and the coastal ocean via breaching of new inlets, storm overwash, migrating inlets. However, our human infrastructure has now prevented most of that from occurring. So these hypothetical experiments are aimed at establishing a controlled connection between the coastal ocean and the Indian River Lagoon, specifically Banana River, to promote flushing. Next slide. And so the tool that I'm using for these hypothetical experiments is the Environmental Fluid Dynamics Code, might be known to some of you, supported by the EPA. The model domain extends all the way from Ponce Inlet down to Fort Pierce Inlet area. And actually this model is nested in a basin scale model called HYCOM, which is the Hybrid Coordinate Coastal Ocean Model, or ocean model, and that provides boundary conditions on the seaward side of this EFDC model in terms of water level, salinity, water temperature. And so I've got about 10 or 11,000 cells in this. And I don't have time to explore details, but let me just show you one example of hypothetical experiment. Next slide. I'm introducing in the model experiment 10 cubic meters per second of inflow in a narrow section of the barrier island just north of Patrick Air Force Base. The hypothetical pump operates for about 12 hours per day. And you can see where it fits in the sort of the model domain of Banana River at the north end of the IRL system. And so let's see what the result is of this hypothetical pumping experiment after the end of a single year. We're tracing the flushing by introducing 20 parts per thousand of numerical dye. Next slide. And here we go. Here's the existing configuration on the left after 365 days. And then the model experiment that includes the pumping station after 365 days, 2016, actually it's 266. And you can see that there's a fair amount of flushing. I introduced the numerical dye for all of the IRL system. But as usual, when you do these experiments, and as we know from the real world, the Banana River is poorly flushed, and the 10 cubic meters per second does provide about 30 to 50 to 60% flushing, if you will, removing that much of the concentration, but still leaves quite a bit. And you can see that mixing zone down at the southern end of the Banana River where it connects at Dragon Point to the main IRL. So it does show that we could introduce coastal ocean water and promote some flushing, although other experiments provided even more flushing, introducing water at the northern end of the Banana River. Next slide. So the phase that we're in now, in addition to looking at the flushing experiments, the rate of flushing, or residence time, if you will, using the water quality component of EFDC It's called EFDC HEM3D for hydrodynamic eutrophication model. And you can see that I have in this particular model, which is fashioned after the Chesapeake Bay model, but adapted to a tropical environment. We have 21 equations of state plus macroalgae, and it's tightly coupled to the hydrodynamics. So we're going to look at the potential water quality benefits of introducing episodically water from the coastal ocean into the Banana River. And that's it.
All right, Gary, thank you so much. Moving on to Austin to tell us about biogeochemical responses to ocean inflow.
So thank you, guys. Can you hear me just fine? Good. So we should not view ocean inflow simply as an exchange of water, but rather a process that will drive small changes to the conservative properties of lagoon water, so namely temperature and salinity. So you can see here on the TS diagram on the right that during summer, lagoon water is roughly a degree warmer and roughly 10 parts per thousand fresher than seawater, and it's quite a bit denser, and that drives some interesting circulation. So changes to temperature and salinity would be proportional to the volume of inflow, and there would be some direct discharge of nutrients. And so this would help to decrease nutrient concentrations in the lagoon due to the 3 to 5 times lower nutrient concentrations in seawater. And so we'll also see a decrease in suspended solids, turbidity, that will help decrease dark respiration rates, or the rate at which dissolved oxygen is consumed from the water column, making the lagoon more resilient to anoxia and fish kills. And so preliminary data shows that the respiration rates in the lagoon are about 3-fold higher compared to the adjacent seawater. So unlike temperature, salinity, and DO, nutrients and dissolved oxygen do not behave conservatively. So small changes to temperature, salinity, and DO can have large impacts on nutrient cycling in the lagoon. And these benefits could be obtained without fundamentally changing the IRL. So in other words, working within the natural range of temperature and salinity already experienced throughout the lagoon, we can see some pretty impressive benefits. To study how changes to temperature, salinity, and DO influence nutrient cycling, we carried out a series of field and laboratory experiments using the sediment incubation chambers pictured here to manipulate the temperature, salinity, and DO. Next slide, please. So inflow of cooler seawater will help to slow biogeochemical nutrient cycling and promote ecosystem services such as denitrification. And so simply decreased temperature equals decreased nutrient fluxes. So experiments also reinforce the literature and show that increased salinity also results in lower nutrient fluxes. So I don't have time to get into that today, but if you want, send me a message in the chat. Inflow of this high density seawater would also help circulate bottom water in some relatively poorly circulated areas now, or chronically anoxic areas throughout the lagoon. And this process would help to maintain oxic surface layers of sediments where nutrients would be sequestered with inorganic phosphorus sorbed to oxic sediment particles and nitrogen oxidized to nitrate and subsequently reduced to N2 gas that leaves the system. So in other words, increased and stabilized DO can decrease internal loading of nitrogen and phosphorus. Next slide, please. So for example, field experiments captured exactly this during recent fish kills. When sediments and overlying water became anoxic, even sandy areas switched from a sink to a source of phosphorus, if you look at that picture at the top left. And so collectively, changes to temperature, salinity, and DO that could result from inflow would likely decrease internal loading of nitrogen and phosphorus to the lagoon. And so when applied to the large surface area of lagoon sediments, both sand and muck, decreased fluxes and decreased internal loading of nitrogen and phosphorus would likely be greater than the benefits achieved just by the direct discharge. And so because DO at the sediment water interface is so important towards regulating nutrient cycling, we've started to deploy our oxygen sensors. So hopefully we're able to catch Tyler's talk or his poster that talked about and introduced that sensor network. So if you have any questions, let me know and help us support and grow this network if you're interested in collaborations. Thank you.
All right, thank you, Austin. Next up is Sean, who's going to tell us a little bit about IRL benthic communities.
Hello, everyone. Hopefully you all can hear me. I'll do a quick sound check. Perfect. Good afternoon, everyone. Today I'll be giving you a brief update on the first year of benthic monitoring done by the Johnson Lab for the Restoring Lagoon Inflow Project. So over at the Johnson Lab, we're responsible for describing the biological communities at these proposed crossover locations, and we're monitoring both in the lagoon and in the nearshore Atlantic. Specifically, we look at seagrasses and macroalgae. We also look at benthic fauna, and benthic fauna is including both epifauna and infauna in this case. We also do examine phytoplankton, but for the scope of this very brief talk, I'll just be focusing on the benthic data. On our right, we have a map of our 3 study locations for reference. We have a Banana River North site near the port. We have a Banana River South site just north of Patrick Air Force Base. And we have a Vero location, which is at Bethel Creek. Next slide, please. Here we have data for an average sampling transect for benthic vegetation. We have seagrasses are on the left and the rooted macroalgae caulerpa is on the right. And it's important to note that both of these vegetation types are recorded simultaneously in each sampling transect that we carry out. Only the seagrass Holoduli wrightii was found during our first year of sampling, and seagrass cover was highest at our Vero location, compared with low and variable cover at Banana River South and low but more consistent coverage at Banana River North. For caulerpa, the caulerpa cover was greatest at our Banana River South location, and there was moderate coverage at Banana River North, but both of these locations exhibited large variability in their cover. And there was virtually no caulerpa found at our Vero location. Next slide, please. And then here we just have some data for total benthic fauna abundances on the left, and we have species richness on the right. Something to note here is that one of the major comparisons is now between our inshore and offshore habitat, and it's less between the actual sampling locations. Something else that's important to note is that the benthic communities of these different habitats are likely to differ in composition. Both abundances and richnesses were higher in the inshore habitat compared to the offshore habitat in the Banana River, and that's for both Banana River North and South. Vero shows a slightly different trend where offshore and inshore habitats are more similar. And this is likely due to the large amount of fine-grained organic rich sediments found within the sampling area of Bethel Creek itself, which harbor very few infaunal and epifaunal organisms. More sampling is going to be required throughout our second year to establish a truly concrete biological baseline for these locations. And then these baselines will then be used to detect possible changes in the benthic habitat of any of these locations if and when ocean water is pumped across. That'll do it for me. Thank you guys for listening. I appreciate it.
Thanks, Sean. Our next speaker is Jesse. He's going to tell us about abiotic determinants of IRL fish community structure.
Thanks, Zach. Is my microphone working while it's pulling up? Awesome. So we've been kind of going through the RLI program, and my team's component is just looking at the fish. Big question, what responses, if any, do we expect from the fish community? And for our first year, I wanted to take just this 62-mile view, zoom all the way back out to space, what's going on, so that in subsequent years we can key in on the structure a little bit more. Question 1, what are the abiotic determinants of our fish? 2, does the community significantly vary with differences in abiotic conditions? And if so, then 3, which are the species driving that pattern? Zach, we'll do next. So thankfully, FWC agreed to provide us with, by my estimate, the single best and most comprehensive fish data to have ever existed. And I've worked with many data sets. Rich's is amazing. So thank you, Rich Paperno. And I tackled these questions using those data in the 3 sites that the previous speaker mentioned, as well as at a global scale, using a series of multivariate analyses. And basically what we found is, potentially no surprise to anybody here, dissolved oxygen, pH, salinity, and temperature are important. That's what it means to be a fish. But what's cool about this is that it's a particularly weak signal. So now we need to focus on the behavioral effects. Question 2, yes, if we were to break these abiotic determinants up across the range that they've been observed, low to moderate levels tend to have the greatest level of structural diversity within the fish community. And then the ones that are driving this signal, question 3, tend to be the low to mid trophic level species. Anchovies, gobies pop up a lot, but then we also see some more commercially important, recreationally important species, red drum, snapper, sea trout. So then, Zach, next slide. So what we're doing with this moving forward then, now we have an idea of kind of these coarse scale patterns of what we've been looking at. For year 2, what we're working on right now is kind of what I'm calling the ground view, developing predictive models of what ended up being 8 species of interest to this particular program, seeing if we can't model their abundance with respect to those abiotic characters, and then forecasting how that abundance might change using the hydrodynamic models and the other abiotic bits of information being developed by the rest of the team, and seeing if we can't predict changes in community structure along the way as well. And then if we're approved for funding into year 3, we're going to be taking this into a much more detailed mechanistic predictive framework of species distribution modeling to get an idea of how the habitat itself is going to change with respect to these species, but also we're going to be keying in on the behavioral component, seeing how fish move and how they react to changes in inflow. So I'm not sure how I'm doing on time. I'll just take the last minute to say thank you again to Richard Colonel, Doug Adams, and FWC for the data, our legislative supporters, Senator Mayfield, Representative Altman, and of course our funding FUAE. And feel free to drop a question in the chat if you have it, because I know I burned through that pretty quick.
All right, thank you, Jesse. And our final speaker of this year's symposium is Jeff. He's going to tell us about an eDNA-based biodiversity assessment.
Great. Thank you so much for that, Zach. I'm really pleased to have this opportunity to share a little bit of the work that we're doing as part of this larger project, Restoring Lagoon Inflow, or the research into the feasibility of restoring lagoon inflow and the practicality of it. What I'm doing as part of this larger project is collecting water samples and assessing environmental DNA within those water samples to better understand the biodiversity across the lagoon, which are candidate sites for pumping, and to help establish a broader baseline for understanding the impact should pumping be initiated. For those of you not familiar with eDNA, just a quick description. eDNA is the detection and identification of DNA that's been passively shed by organisms present or recently present in the sampled habitats. And usually the substrate is water samples or sediment samples, but principally it's most often water samples. And then the DNA is filtered out of those water samples and then examined and identified. You can see here from figure on the left that the interest in eDNA has been increasing rapidly over the last several years. And we have two approaches, single species and multi-species approaches. I'm not going to go into the details of either of those approaches, other than to say that the multi-species approach, the metabarcoding, is what we're using in this case. And it has an opportunity or creates an opportunity to assess whole communities, or at least to get taxonomic data or diversity data across large taxonomic groups. Next, please. And so eDNA itself is more commonly used now, or is being increasingly commonly used because it offers non-invasive sampling. It's a complement to traditional sampling. We can sample water or sediment in areas that you often can't conduct traditional surveys. And then because of the broad taxonomic representation that I just mentioned for the metabarcoding approach itself. Next. Thank you very much for that. Here, what the goal is to look at or increase our understanding of the occurrence of fishes and invertebrates within the potential sites for this proposed pumping project, and then adjacent habitats as well, in order to better understand the impacts of pumping should it be initiated. And then with this data, we'll be working up species richness and distinctiveness to be able to actually better assess how the system might respond to this particular management intervention, if it's actually initiated, as well as ongoing and proposed other management strategies. Next, please. So for the eDNA sampling, we have 14 lagoon sites that we've sampled in November 2019 and in February 2020. And then COVID hits, and we haven't been out to sample across the IRL since the initiation of COVID. We're able to get in an offshore sample as well that's complementary to our initial inshore sampling in February 2020. We have sampling coming up soon in March that'll be across this same area with the exclusion of the offshore sites. And then again, late spring and into the summer as well, assuming we can get an extension or some alteration of our funding term. One of the limitations of this project right now is that just the funding cycle itself leaves us challenged to conduct the research in the summer, which, as we know, with the seasonality of species within the IRL, it's a pretty important season to be out sampling and assessing diversity. And we want to be able to include that in our study. Next, please. So we've taken a multi-gene approach to this eDNA, to studying eDNA. We use CO1 gene and 16S gene. The idea of the CO1 gene was to provide a broad assessment of diversity across all metazoans. One of the challenges we find with this broad assessment is that the primers needed to be able to amplify so many different species are fairly universal, and then they end up picking up a lot of harmful algals and other present single-celled eukaryotes that they complicate an assessment of the diversity for the target species we're most interested in, and they make the utility, right now, they undermine the utility of the CO1 gene in this case. We've also are using 16S to provide more detailed information on fishes. Is that it's actually, because it's a little bit more targeted approach to fishes and specifically, we have better luck using it during or even on the outside edges of harmful algal blooms, which we've actually done some sampling recently right in the middle of the bloom that was occurring up in the Banana River in late fall, and found a number of challenges that need to be overcome to be able to apply this technique in the middle of a harmful algal bloom. And we're working around those right now. So based from the 16S data, what we saw is a total so far of 27 families that we've identified. This is a preliminary first pass assessment of our sample diversity. And then within those 27 families, we have 35 species actually assigned to species level. We have other taxonomic assignments as well at higher level that offer some information about diversity patterns. But here I'm presenting just the family and species level, as that's just the most informative. And you can see that the data on the families in the figure on the top left there, with the numbers in parentheses of the actual numbers of species that were sequences that were assigned to species level, or taxa that were assigned to species level within that group. And then you see on the figure on the left in the center there is a heat map of the distribution for all of our taxonomic assignments. And just really quickly, you can see that there are some geographic patterns to the distribution of these species. So we are picking up this spatial distribution of species within the IRL with this eDNA approach that I think with more sampling, we'll be able to actually resolve quite well the regional patterns of species diversity, as well as the seasonal patterns. And so next, please. And so with that, I want to mention, as I'm the last talk of our little mini series here on the Restore Lagoon Inflow project, I want to mention that this is a large project, including a multidisciplinary team looking at hydrodynamic patterns, geochemistry, quite a few aspects of the biology of the lagoon. We're working with a number of partners to be able to expand understanding of the lagoon and the trends in the lagoon, and our ability to actually predict how the inflow is going to affect all these components. This work is sponsored by Senator Debbie Mayfield and Representative Thad Altman, and it's a direct response to increasing public concern about declining lagoon water quality. So this research that was sponsored by the states is examining aspects of lagoon water quality and how we might be able to better manage lagoon water quality. The work addresses specifically 4 IRL, NEP, CCMP vital signs, including one that specifically supports investigating the potential benefits of enhanced inflow. And we see this from the beginning. We've seen this as a potential tool in a multifaceted approach to managing the water quality in the lagoon, not as a single approach that's going to solve all of our problems. But
Hey, Jeff, the giant net's about to come out from behind the curtain.
All right, let's go click one more. One more click, please. I just want to say we're in the middle of our second phase right now, building off of phase 1 results. Phase 2, 3, and 4 are a pilot project proposed for Port Canaveral. Right now, we're in discussions with the U.S. Army Corps of Engineers and Port Canaveral to initiate this project, and with our state funding support. And we're more than happy to share any of the results, or if you'd like more information on this project, I'm going to post a link again in a minute. And I want to thank you, everybody who's helped with this project in the past and ongoing. Thank you very much.
All right, Jeff. Thank you so much, and thank you so much to all of this year's presenters, particularly our student presenters. I think the symposium was a huge success, and I'm going to turn things back over to Dennis to wrap up.
Thanks so much. It was Very good. This will not take anywhere near the time that is listed on the program. I'll be very quick. A couple of reminders. Don't forget to vote for the People's Choice Award. You've been advised that a couple of times in the program. Also check the chat. We will close that at 2:30, so we'll give everybody an hour. Take your time if you need the time. We'll send a global note on recordings of the oral sessions once we have it posted. A number of people have asked about that, so we'll take care of that. I don't know about you all, but I've been thinking a lot about this year's theme, IRL research in the pandemic. I think we've had many great presentations that address that kind of at project levels. What I've been wondering about is there anything else we may have learned at a higher level, perhaps? For example, is there anything we can learn by watching this whole pandemic unfold? Can we apply it in River Lagoon? Perhaps it's this. I think it's the importance of excellent science that solves a crisis, whether that involves a virus or a lagoon. And another important ingredient for solutions is broader collaboration among all parties involved. So let's think about that more in 2021. And hopefully we'll still call what we're going through now the pandemic as opposed to the first pandemic. So let's hope we never have to do this again. At the opening yesterday, I explained our dilemma, whether to do IRLS 2021 virtually and why we decided to proceed. I think we all know that virtual meetings are challenging, but maybe there are some things better about a virtual meeting than an in-person one. So I've been trying to think of that too. Here's 3 ideas. Unless you are presenting, you don't really have to dress up. And then only when your camera, you only have to address what your camera can see. Another thing about this meeting is you don't have to wait in that long line with your parking pass trying to get through the Harbor Branch gate in time to get a coffee before the first talk. And no long drives home. That means that everybody can attend the Student Presentation Awards. Don't forget the IRL Trivia Challenge and the Student Awards at 3:30. I hope to see you all then. And next year, I hope we're all together again. And I hope unmasked. We're going to keep this room open for a while. If there's any further chat among yourselves, that'd be great. Judges, remember to send in your information via the designated pathway. If you have any problems, you can send them to me. I've kind of been the conduit for anybody who's having a problem, so I can continue to do that. Thanks. Thank you all. Appreciate everybody who helped in any way, shape, or form and everybody who attended. Look at that. Totally within my time limit. See you next time.